So, Prof, you can start now.
Okay.
You can go ahead and start. Sorry for the confusion.
He is muted. He is talking but muted.
Oh, okay, okay. Please, Prof. I think you are muted. Take this elsewhere.
Good morning, everyone.
Good morning.
I'm very happy to be with you today. And I'm going to give a short talk about natural products in modern medicine. The topic today to be discussed will be mainly discussing three main points. Number one, what's the importance of natural products? Number two, role of natural products in modern medicine. Number three, future of natural products in drug discovery. We are just going to discover the mine of natural products, or gold mine of natural products, and the role of natural products in modern medicine. So we are just going to just take a submine and going to the treasure under the sea. Why natural products are important today? Role of natural products in modern medicine, future of natural products in drug discovery. Number one, why natural products are important? According to the World Bank report, global market value for herbal industry was $100 billion in year 2004. United States is controlling $60 billion from the $100 billion. Prescriptions containing phytochemicals in the United States were valued at more than $30 billion in United States 2004, from the $100 billion around the world or globally. If we are going just to investigate the prescriptions around the world, currently 1/4 of all prescribed pharmaceuticals in industrialized countries contain compounds that are from natural sources or plants. If we are going just also to see the preparations around the world from herbal sources, we are going to find most of the preparations, or more than 95%, are whole extracts. Whole extracts like ginseng extract, like valerian extract, total chamomile extract, total hops extract, total Celebium marianum extract, because the active constituents will be synergized by some other constituents present in the total extract. Like as an example, in 2004, Novartis company just industrialized a preparation called X-Lax, which contains sinusides only. The sinusides are the main constituents present in senna leaf. When they were trying this preparation, they found that this preparation is 1/4 of the activity of the total senna extract, because there are some alloimodine compounds which are not biologically active, but they will activate the absorption process or the bioavailability of sinusides, and also decrease the metabolism of sinusides. So they are synergizing the bioactivity of sinusides to 4 times. So most of the preparations of the herbal preparations in the pharmaceutical markets are in total extracts as a general. The WHO is saying that more than 80% of the approved drugs are either natural products or related to them, including biologicals such as vaccines and monoclonal antibodies. Natural products are the most important anti-cancer agents. They are saying that 131 compounds out of 175 of the anti-cancer drugs are natural, like as an example, Taxol and its derivative. Also, the anti-infective agents like cephalosporins and beta-lactam compounds are from natural sources, from certain types of fungi. Nature is still the main source for pharmaceutical compounds like streptomycin. Streptomycin was discovered in 1943. It's an antibiotic. Was discovered by the American scientists when they were seeking for a drug used for treatment of TB during World War II. But the streptomycin has side effects upon injection. It's going to affect the optic nerve. So it's now used locally in the GIT infections. Also, doxorubicin is an anti-cancer drug used till today. It was discovered in 1968 from certain types of fungi. It's used for lung cancer and also breast cancer. It's from natural source also. Role of natural products in pharmacy.
Professor Sabri, your slides are not changing because we are on the first slide of natural products in modern medicine. Why, role, and future? That's where it is stuck. Is it moving from your side?
But now it's changing.
Please. Reshare again and see what happens, huh?
Okay. So now it's working or what?
Just check, share again. So otherwise you can also email it to me and I can drive it from this side. Let me give you, in case of anything, let me give you my email in the chat.
Okay.
Sometimes Zoom refuses to share properly. No, sorry. So, let me share to you. So there's a message that has come with my email. And if it is, yes, okay, now we can see it. Just see if you can go, yes. So now can you start maybe closer to the beginning because your slides were stuck on the first slide, huh?
Yeah.
Okay, good, thank you.
You are welcome, So again, it's according to the World Bank report, global market value for herbal industry was $100 billion in year 2004. United States is controlling about $600 billion from this value. Prescriptions containing phytochemicals around the world were valued at more than $30 billion in the United States only in 2004. And the United States is exporting more than $30 billion as a dietary supplements or something like that. Currently, 1/4 of all prescribed pharmaceuticals in industrialized countries contain compounds that are from plants. If we are going to see any preparation present in the market, we will see that they are enclosing the whole or the total herbal extracts. Like as an example, ginseng extract, valerian extract, chamomile extract, hops extract, like that, and also milk thistle and dandelion extract. Why they are using the total herbal extract? I'm going just to give one example. Novartis company prepare just a preparation of pharmaceutical preparations or pharmaceutical preparation, which was X-lax. It contains sinusides from senna. When they were trying the biological activity as a laxative, they found that the activity is 1/4 of the total senna extract. Because they found after that there are some bio-inactive compounds like alloimodine. These compounds will elevate the number one absorption of sinusides. Number two, decrease the metabolism of the sinusides in the body. So they will synergize the activity of these sinusides inside the human body. So they returned again to the whole total extract of senna to be used as laxative. And this is applied for all of the herbal preparations, almost. The WHO is saying that more than 80% of the approved drugs are either natural products or related to them, including biological, such as vaccines and monoclonal antibodies. Natural products are the most important anti-cancer agents, like Taxol and vincristine, vinblastine, camptothecin. They are saying also that 131 compound out of 175 anti-cancer drugs are natural. Even the new compounds are modified natural compounds like Taxol and docetaxel, which is used now in breast cancer. Anti-infective agents like beta-lactams and cephalosporins are from natural sources. Cephalosporin C was discovered in 1948, and today it's the main pharmacophore. They are modifying the side chains, but they are producing modified compounds, second generation, third generation, fourth generation, but it's from cephalosporin C. Nature is still the main source for pharmaceutical compounds, like streptomycin. Streptomycin was discovered in 1943 when the American Army was searching for a drug used for TB because soldiers were infected with the TB microorganism. They discovered the streptomycin, but streptomycin upon injection, it has a side effect. It's going to affect the optic nerve. So they are using today streptomycin as a local antibiotic, especially in GIT infectious diseases. Doxorubicin also was discovered in 1964. It's from natural source, and it's used today in lung and breast cancer.
Sorry, we lost your slide, Prof.
Yeah, I'm going just to take the third slides or the third part of slides. Now it's a new slide or what?
We are on
Ivermectin,
yeah,
E.
yeah.
A Japanese scientist noticed that some clay contains no worms. He was trying just to discover the reason. He just isolated a type of a sponge or a streptomyces sponge, streptomyces fungus, I'm sorry, and he got this streptomyces and left to United States and worked with Dr. William Campbell. Both of them got the Nobel Prize after that. From this fungi or fungus, they isolated a compound, macrolide compound, which is a very strong halogen or chloride, causing chloride influx inside the neurons of the worms, especially. So this is going to cause paralysis. So this is why this clay contains no worms. They discovered this compound and they got Nobel Prize, and it is now this compound is a drug of choice for treatment of filariasis. And also it was used last 2 years for COVID-19. Biguanides, they were discovered from Gallica officinalis. This is a plant present in Tunisia, Morocco, and Algeria. When the French scientists in 1930s, they just got this plant and they discovered an alkaloid which is called galligine. This galligine, they discovered that it can treat polyuria. Also, this plant was used in treatment of polyuria in the African countries by folklorical use. After that, they say it's a hepatotoxic compound, but we can modify that just the side chain. It changed to biguanide and then to metformin or metformin, which is pseudophage or glucophage today. So glucophage or metformin was discovered from a plant from Morocco and from Tunisia by a French scientist. And the first company just prepares this preparation. Metformin was a French company also. Lovastatin, which is used as anti-hypercholesteremic. There was a Japanese scientist from Japan. He said, I am ambitious and I want to get Nobel Prize. He said, we have in Japan just what's called red yeast. Red yeast is used as anti-hypercholesteremic and anti-hyperlipidemic folklorically. He was a biochemist. He just investigated the cycle or the biosynthetic pathway of cholesterol from acetyl-CoA. He just discovered a compound. He left to the United States for a PhD, and he was working on red yeast. He isolated some compounds. These compounds were active against the biosynthetic pathway of cholesterol. It is going to stop this cycle in a beta-hydroxy beta-methylglutarallyl-CoA reductase. This enzyme is blocked by the compounds that was isolated by this scientist. And then Merck company in 1985 just isolated from also red yeast the lovastatin compound, which is Mivacor, and it was the first compound to be discovered for treatment of anti-hypercholesteremia from nature. It's a statin, one of the statins. Also, a Chinese scientist, Dr. Tu Yoyo, she got a Nobel Prize also for isolation of artemisinin from Artemisia annua. Artemisinin is used for treatment of malaria by degeneration of the heme, oxidation of the heme. So the nutrition of the heme is not now of a value for this microorganism. Reserpine alkaloid discovered in 1952. This compound depletes. This compound depletes the vesicles containing catecholamines from the pre-synapses, so it's used for treatment of hypertension. But a side effect, and Novartis company just manufactured a compound called Brenardine. But they noticed that the patients were trying to suicide after that, some patients. They said, so reserpine is going to deplete the catecholamine vesicles, so the catecholamine level will be decreased in the brain. It's causing depression after that. So they discovered that increasing the catecholamines in the brain can treat depression. So this was the first reason or cause for the discovery of MAO inhibitors as an antidepressant. And the first compound was discovered or synthesized, phenelzine, by Pfizer company. So reserpine was the cause for the discovery of MAO inhibitors as antidepressants. Also, clavulanic acid. Richter company from Denmark, just they were trying to isolate an antibiotic from Streptomyces species. They isolated cefamycin and also a compound called clavulanic acid, but they found no activity for the clavulanic acid by itself on the bacteria. But when beta-lactam resistant antibiotic problem appeared in 1985, they go again to clavulanic acid just to add with amoxicillins for beta-lactam inhibitors as general. So it's used to synergize amoxicillin against the beta-lactam resistant microorganisms. Pfizer company in 1985 were trying to discover a drug used for anginal attacks just as a vasodilator from purine alkaloids or xanthine alkaloids like theophylline. When they were trying, they discovered a compound. This compound is called sildenafil. They were trying to discover a compound for treatment of anginal attacks. When they were trying in the clinical trial this drug, they discovered that it can treat erectile dysfunction. And Pfizer company approved this compound for treatment of, as Viagra, for treatment of erectile dysfunction as a phosphodiesterase inhibitor, phosphodiesterase 5. We have to know we have phosphodiesterase enzyme 1 in the brain, number 3 in the lungs, number 5 in the erectile area. So it's affecting or it's concentrating in the erectile area. So it's a phosphodiesterase inhibitor in this and causing vasodilatation and increasing blood flow, increasing blood flow. So it's used in treatment of erectile dysfunction. The chronotype, there was a Filipino doctor. He was working in a Utah university in the United States. He just noticed that when he was a child, he was taking a snail. It's called coon snail. This coon snail, when any fish is going to attack this snail, is going to sting this fish and injecting a poison. This poison is going to paralyze the fish, and this child is going to pick up fish easily. He was saying, why is this coon snail is going to paralyze this fish? And what's the chemical compounds that it's secreting? They started to work on this coon snail, and they isolated many compounds, many polypeptides called conotoxins. The most active one was what's called omega conotoxin. This omega conotoxin consists of 27 amino acids. This compound now today, it's converted to a drug used for treatment of bone cancer. It's called ziconotide, and one vial is $7,500 each vial. Today they synthesize this compound. Natural products here, if we are going to look for acetyl salicylic acid as an example, the source of discovery of acetyl salicylic acid or aspirin by Bayer company was salicine, which from Salix purpurea or Salix alba. Synthetic salicylates produced by Bayer company in Germany, which is acetyl salicylic acid here. The cocaine was used as a local anesthetic in old times. Cocaine was a model for the local anesthetics generally. If we are going to see, so cocaine is the parent of the whole local anesthetics used today, because just it's a modified pharmacophore or motif compounds. Also, the ACE inhibitors that we are using globally are from natural sources. If we are going to see, there was a viper in Brasilia, or in Brazil. It's called Brazilian viper. This viper, when just attack, it's going to attack anyone. It's going to just cause severe hypotension. So Merck company, with the help of some Brazilian guys, just start to discover what's that. They discovered that this viper venom contain what's called tebrutide. It's a nonapeptide, contain 9 amino acids. They discovered that the pharmacophore here is the proline amino acid. So they started to work. This is just angiotensin 2 converting enzyme inhibitor, or ACE inhibitor as a general. All of us are knowing that there is what's called angiotensin is going to be secreted from liver, and it's going to pass through the kidney. By renin is going to convert it to angiotensin 1. Angiotensin 1 is going to, in the lungs, is going to be converted to angiotensin 2. Angiotensin 2 is going to control the blood vessels. Upon presence of angiotensin 2, blood vessels are going to contract, increasing blood pressure. When we are going to suppress the angiotensin converting enzyme in the lungs, so for sure we are going to treat hypertension, and this is the mechanism. And the first compound was discovered in 1985 from the proline, was by Merck company, which was called captopril. So it's from natural source. And also there is a second, third generations from these ACE inhibitors. Also, we can see that today, natural products are serving as extremely useful drugs like vincristine for treatment of leukemia, vinblastine for lymphoma, Taxol for lung cancer, digoxin for congestive heart failure, atropine as a spasmolytic, ergometrine for postpartum hemorrhage, morphine as a narcotic analgesic, papaverine as a smooth muscle relaxant. Also, natural products can provide new semi-synthetic compounds. Like some scientists tried to modify the structure of vinblastine, which was used for treatment of lymph cancer, and they discovered new anti-cancer drugs like vendazine, which is used for melanoma, vinfluline for bladder cancer, venrolibine for breast cancer. Modification of Taxol side chain as an example gave what's called docetaxel. Taxol is used mainly for lung cancer. Docetaxel is going to concentrate in the breast, so it's used for breast cancer. Also, modification of bodofolio toxin gave what's called etoposide. Etoposide is used as a topoisomerase B inhibitor, and it's used for lung and testicular cancer. Also, we can use natural products from inactive form to be converted to active form, like what's called diagenin. It's present in fenugreek and wild yam. We can convert these compounds to progesterone, estrogen, which are used in contraceptives, and also we can convert them to cortisones used as antihistaminics, anti-asthmatic. We can go to the fourth slides or fourth part of slides. And it's I'm going to the end within about 2 or 3 minutes. Today, there was, as an example, a problem in the supply of Taxol. Taxol, if you want to treat a person with breast cancer, as an example, or lung cancer, we have to use 2 grams for this person from Taxol. So we need 6 trees with 100 years old trees, and also we need 6 trees to get 2 grams. So this was a problem. So they tried first to produce Taxol by cell suspension culture, plant cell and tissue culture, and then from 2019, they discovered the gene cluster responsible for the biosynthesis of Taxol, and now they transfer the gene cluster to yeast. They are doing like what was done in insulin, humanized insulin. So it's now available, Taxol, and I'm sure it will be more cheaper. So now today, if we have any problem for the supply of natural products, we can use the modern techniques. We can use plant cell and tissue culture. We can use plant biotechnology or biotechnology as an example. Also, if we have some problems with some bioactive compounds, we can use the drug targeting techniques today. As an example, if we have Taxol has many side effects. If we are going to target Taxol to the cancer cell only, so we are going to get rid of many side effects. And this is what is done today. We can, but these preparations are very expensive. We can use gold nanoparticles, and also we can use silylated polyethylene glycol and silylated compound, which is Taxol, and tumor necrosis factor, which is this is the main leader to the cancer cell. We are going to link all of these compounds with each other, and you can see just a video after that. So we can get rid of any side effects from Taxol, like that. So we are going just to take the drug and making linking with gold nanoparticle, silylated molecule, and the antibody for the cancer cell. So this antibody is the leader for this complex to the cancer cell because it is specialized or specific for this type of cancer cells. So when the antibody is going to link to the receptor on the cancer cell, it's going to enter by endomitosis. And then the drug will be evolved, or the protein of the antibody will be lysed by proteasomes. And the drug will enter to the cell specifically, and it will make suicidal or apoptosis. Today also we have many compounds, like I am sure that we have more than 350,000 natural compounds, most of them without pharmacological activity or without known pharmacological activity. We can today, using the computer-aided drug design, just rediscover these compounds to investigate if they are working on certain receptors or certain enzymes. So we are going to discover also these non-discovered natural products. Also today, from 50 years, if I want to carry out structural elucidation of any natural products, I need at least 100 milligrams. But today, with nanograms, I can isolate and I can confirm the structure of the compound, and we can synthesize it after that. Or we can even just transfer the gene cluster responsible for the biosynthesis of these compounds. Today also, by knowing the mechanisms, we have many. Today, we have what's called pharmacologically or pharmacological networking. If I have some structures or some compounds, I can give the software, the compound, and it's going to predict the pharmacological activity of the compound. Like as an example, there is a type of mushroom which called white destroying angel. It's a mushroom containing alpha amanitin. It's a polypeptide which is causing RNA polymerase inhibitor. So it's going to inhibit the RNA synthesis inside the body. So it's toxic. But if I'm going to use it for certain microorganisms, it can be used as antiviral. Also, it can be used in treatment of certain types of cancer. This is just a snapshot from the role of natural products in modern medicine. Thank you. Thank you.
Thank you very much, Prof. So, Sameh?
Thank you, Dr. Omar, very much for your very informative and interesting presentation. I'm sorry I was not here in the morning. Now the floor is open for discussion, please.
So let me start. Thank you very much, Prof, for your nice presentation on the role of natural product as a source of drugs. You have also discussed about the new technologies that are available to bring even tissue culture, computer-aided drug design, from poison to drugs, etc., through card studies. So most of, nature has been a very good source of single bullet drugs like artemisinin, like Taxol, etc. But in majority of the cases, as we have also discussed, the single compound, when a plant that is found to have very good biological activity, when it is purified further, it loses its activity, and as a result, the whole plant or some extract are used in phytomedicine. I just want you to tell us, if possible, what is really something special about some of the drugs like Taxol, artemisinin, for them to become a single bullet drug, just like what we have in synthesis. I don't know, do you have any idea about what is special about those few examples, like chloroquine, for example, artemisinin, Taxol, vinblastine, vincristine, which as a single compound have very good pharmacological effects, but for many plants, when they are isolated, when they are separated, they lose their activity. Thank you, Prof.
Yes, very nice question. We are looking for specificity first. So if you have vinegar alkaloids as an example in a mixture, so they are going to distribute in different organs in the body. They are going to work as antimitotic. So they will have a lot of side effects. So we are going to choose vincristine just for treatment of leukemia because it's a polar compound. It's going to concentrate in blood. So it's used in treatment of leukemia. Vinblastine is going to concentrate in lymphoma. So it's going to use for treatment of lymphoma or Hodgkin's disease. So we are looking for, as an example, if the alkaloids of ergot. Ergot alkaloids, ergot contain more than 65 alkaloids, like as an example, ergometrine, ergotamine, ergonovine, ergotoxin. If I'm going to give a lady in a labor, just the whole ergot extract, ergometrine is going to work just for the treatment of hemorrhage, but we have side effects for the other alkaloids because I want to use it for a specific use. So ergotamine is going to work as a vasoconstrictor. So it's going to may cause gangrene or very high blood pressure or crisis in blood pressure. So I cannot use a total ergot alkaloid in this case. Yeah.
Yeah, now the floor is open for discussion again, please.
Yes, first I would like to thank Dr. Omar for a very good presentation. Now my question is regarding to how we can develop our herbal medicines in our countries, because if you look at the average time it takes to develop a pure drug, from the process of drug development until it reaches the market, it takes many years, up to 12 years, 8 to 12 years. While if you are to look at the approach of the herbal medicine, it will take a much shorter process. But typically, to come up with a new drug for the big pharmaceutical companies, they may spend up to $800 million before they do so. Those companies are not going to invest into our natural products unless our own countries or regions are ready to. So I don't know what is your experience in your country, how maybe you can be able to overcome this financial problem. Thank you.
Yeah, this is also very, very good question, but this is a problem in the legalization and the regulations in the Ministry of Health in our countries. If you are going to see in European countries or United States, you will see that they are very easy. So you can just approve your product in a few months, sorry. But this is a problem in the governments or ministries of health in our countries.
I want to stimulate the discussion again with the participants, please, of this summer school. We have my name. Yeah, please, Professor Kamal.
Thank you, Professor Omar, for the nice presentation. My question is similar to the previous question. With this excellent technology and considering the cost of developing the modern medicine, I saw in one of your slides where this drug was changed from the poison of a snake to the drug 1965 to 1981. That is 16 years. So with the economy of African countries and possibly Asian countries, how can we use, what do you think, how can we use this modern technology to develop this African herbal medicine without going into this one molecule drug, which we are almost looking at now as impossible for most African countries? Thank you.
You are welcome. I think I'll be frankly or truly with you. If you don't have economy, so pharmaceutical industry will be very weak. And that is the problem in our countries also. We don't have any defect in scientific backgrounds as a scientist here in Africa, but we don't have the financial support, in fact. This is the problem. And you are going to see if United States is going to take the herbal medicine or herbs from Madagascar and is going to manufacture the compounds. Like as an example, the Venica. Venica was from Madagascar. Who's producing now vincristine and vinblastine? They are not Madagascar country, no. They are the American guys. So, in fact, if you don't have economy or money, you will suffer a lot.
Lost connection with us, I think.
Hello, Prof.
No, he lost connection. Please.
Yes. Yes. Hello, Prof. Can you hear us? Prof, are you with us? Okay, good. Thank you very much. So on our side, the time will break for 20 minutes and come back and continue with our program. Thank you very much.
I'm sorry, the internet was disconnected. I'm very sorry.
Oh, okay. You are back?
Yeah. I'm very sorry.
Yes. So Thank you very much, Prof. Please clap. Thank you very much. Thank you very much, Prof. We're very, very grateful. You are very resilient. Nothing can beat you. We have made to successfully given us your talk, and we'll continue to engage with you, Prof. Some of the participants will want your presentation, and I've shared with you my email, so you can convert it in PDF and share it with me, so that we can share with our participants.
For sure, for sure. I'm very happy just to share this presentation with you and with the colleagues here.
Thank you so much. So we'll go for tea break and then we come back. Thank you.
So I can come to just to drink tea with you, yeah?
Yes. So we're coming back at 10:30.
Chai.
Was it Safaricom?
No, I'm using the UON Wi-Fi.
Oh, did it work for you?
Oh, it's not working there.
No.
Oh.
Remember that interruption?
Oh, mine is working so well.
Hello, Dr. Tanwi.
twihuse cyane tudatinze rero n'ubundi twongerezwa rifisi muri ibintu bihinduye isura kuko noneho kuri ubu ngubu indege za kajugujugu za MONUSCO zikaba zatangiye guhigisha uruhinjo abarwanyi ba M23 mu kirere ndetse no ku butaka. MONUSCO yiyemeje kuhohereza abasirikare benshi bashoboka mu rwego rwo guha umusada igisirikare cya FARDC ndetse n'abarwanyi ba FDR ubu basubirijwe cyane n'inyeshyamba za M23. Nayo makuru tugiye kuba tugarukaho muri aka kanya, ariko rero namwe nimureba aho hasi handitse subscribe muhakande ubundi amakuru yose mujye muyabonera. Ese mwumva ko M23 ariyo babwira bati mirwana kandi abateganya babwira umuntu niba bahagaritse ngo beko
Hello, we are back again. So now, hello everybody, good morning our online audience. Let's start a second presentation of our course day from the summer school of comstech NAPRECA 2022. And now it's my pleasure to introduce to you one of the great scientists we are all proud to have in Africa, Professor Abi Yansu, a professor of chemistry at the University of Nairobi, Kenya, who has extensive experience in teaching organic chemistry, especially organic spectroscopy, and also conducting research in the field of natural product chemistry for almost 30 years, more than 30 years. He investigated many plants in Africa, especially Eastern Africa, with over than 100 publications in reputable international journals. I also co-authored many books about medicinal plants in East Africa and served as a reviewer for certificate and master. Currently, he's like a subject editor of the very reputable journal Phytochemistry Letters, and serving also as an editorial board member of the Journal of Natural Products and Journal of Pharmacology. Professor Apier, well-known scientist in the world, and we're very proud he served as program officer and assistant secretary for our organization, NAPRECA, and he is one of the leaders of the organization involved in organizing many symposia and workshops at summer schools and coordinating many postdoctoral workshops, promoting collaborative research. The honorable biography, very happy Professor will be with us today. Honorable biography is a fellow of African Science and member of Royal Society of Chemists. I thank him very much for being with us today, and I ask him please to start his presentation. Professor Abiy, the floor is yours.
Thank you, Sami. So it's not it's okay. Okay. Let me see if I share this video. PowerPoint is off. I mean, PowerPoint is on. Where did your PowerPoint? Huh? No, things. I don't know.
Let me tell you, I think the benefit, I can tell you that it can be very interactive with the expert in the field, in the farm, and the international infrastructure. So be prepared for questions, for exercises, a lot of interaction with the Please.
Thank you. Thank you, Sami. Thank you, Sami. Allow me to present while seated. Good morning, ladies and gentlemen. Anything about it? No, I can't. No, it's good enough. Thank you once again. The technological hitch goes with the benefit, so just thank you. Yeah. Well, Solomon is the one who gave me this title, and then he has already decided what I should say. I'll try to fit in the clause he prepared for me. I'm sure those of you who are in natural products science, I would say the most important tool is NMR. And well, I had few years of experience. As you can see, the gray hair. So even if I'm not knowledgeable, I'll share the little I know. Well, I usually prefer to start my talk on this provocative first slide. This slide is the world at night. If you check Europe, North America, to a limited extent, Asia, you'll see it is well lit. In a way, the light also represents the scientific investment made in Europe and America. And it also represents the discoveries, innovation, and also publications. I'm sure you know where we are. They call it the dark continent. Well, if you really look carefully, East Africa, Kenya, and Uganda, there's a bit of light. That light is not fluorescent light, but kerosene lamp. And our investment into science is as good as kerosene lamp. But of course, still use kerosene lamp technology. And especially in NAPRECA member countries, for the last 30 years or so, our mentors who started, we have followed to make sure that even if it is kerosene lamp, we are visible from space. So the story I have within NAPRECA
now in the Department of Chemistry, University of Nairobi. We have a National Product Group for a while, led by Professor Midiwo. Now he has left us for another university, but if the captain has moved, the rest of us have solved their own and we continue. I'm sure you recognize some faces. Solomon and Professor Kerubo, and some of the participants here are also aware, member of our natural product group. But as we try as much to do self-sufficient, but of course, we all need support from up north. The ISP from Sweden has supported us, and also Germany, DFG, and BMZ, through the collaboration created to a group in University of Boston have supported us, and myself, Solomon, and Professor Kerubo have benefited from such collaboration and mentorship. Recently, we also have good South-North collaboration with a Swedish group led by Professor Marta Erdek. And he has hosted and involved in training of a number of our students. And all this, at one point, they have visited his lab and benefited from advanced instrumentation and knowledge from his group. Right now, Ivan and a few other students are in Professor Marta's lab. Well, we all claim to care about the destitute, poor, and sickly African children. They say the solution is always found along the programs. So natural product research, if you want to help these children, solution is also within. We have a diverse bioflora. And of course, utilization of our natural resources requires a multidisciplinary collaborative approach. So collaboration involving pharmacy, biochemistry, biology, chemistry is what we need. And I think that's how even NAPRECA was formed, to create that framework. And thanks to Dr. William Kama and the bodies in this court, I'm sure the collaboration will be more fruitful. Well, our biggest asset is our traditional knowledge. And this traditional knowledge is being exploited by local communities, harvesting, preparing, and we even have local pharmacists, and we have our local medicinal doctors. Such information, I'm sure most of you recognize this book by Professor Kukwar. Any natural product scientist worth his or her salt at one point must have used Professor Kukwar's book. So we call it the Bible of medicinal, the Bible of ethnobotanical knowledge. And even if could not extract direct the knowledge from the users, from the communities, as chemists, in most cases, use such information and carry our plans to a lab and do some research. I'm sure you recognize Huntington years back working in our lab before he has matured and become well, recently we followed what Professor Kukwar, the late, has done in collaboration with Dr. Najma of Kenyatta University. We also wrote a book. I say Professor Kokoro's book is the Old Testament. I can claim this book is the New Testament. Those of you who are Christians, if you have not read the New Testament, you are not Christians. So make sure you get the New Testament of medicinal plants. Why I call this book the New Testament, it's not limited to having traditional knowledge documented. So we have done some work. Part of the work has been incorporated into this book. So we also find the pharmacological reports, chemistry, the active ingredients. After I have advertised our book, now I go back to what we do. Our research objective remains to identify anti-infective principles from plants. So our was mainly on malaria because we have good collaborators. But more importantly, we say we are first generation scientists. So our principal task is to train those undergraduate and postgraduate students so that the next generation of scientists will actually solve our problems, African health-related problems. Myself, of course, our flora is quite wide. You cannot do everything. The last 20, 30 years are focused on 4 plant families. So the Lacea, Leguminaceae, Rubiaceae, and a bit of Rubiaceae, but mainly have done a bit on the phytochemistry of Leguminaceae. Knowing the plants is one thing, but of course, you always have to go to the lab and do the bench work, or we call it the donkey work. I'm sure most of you have spent hours, days, weeks, and years in the lab to isolate compounds. In most cases, the amount of labor and skill put in the isolation process is not appreciated. But of course, in the final analysis, we have to do structure determination. And for a long time, we had a running 200 megahertz. We have used it for over 10 years until the kerosene lamp got extinguished. While even in darkness, without NMR, without light, we continue our research. But of course, the South-South collaboration with biologists, especially with Cambridge, Dr. Akala is the one who did most of our plasmodial assay. But of course, when it comes to structural elucidation, we need high-field NMR, and we look up to the north to look for fluorescent light. Then through collaboration, at least we have bridged the gap. And our 2 main collaborators are a German group and and a Swedish group. In Germany, Dr. Matthias Heidelrich, I think he must be an honorary member of NAPRECA and an African. And I think even his mindset is like an African. When we have worked with him close to 25 years, and he has remained a good brother. And then, of course, also extended collaboration with Sweden, Professor Martha Elderly, another excellent chemist, and very useful. Now he has moved to Uppsala University, where we still continued our collaboration. So at least we get our compounds, we send them, analyze, and then send the spectra to us, and we do the interpretation, and it's collaboration we publish with them. And I think this South-North collaboration has been useful when we get true and useful Northern partner. To give you some examples of what we have achieved, remember it is about NMR. I'll give you some examples of how we have interpreted. We say we mainly train the students, because the students who have done the interpretation. Students we train are as good as any PhD graduate from any other university. In fact, some of our students, when they go to Germany, I'll show you one of them, they used to call him Dr. Spectra, because professor, when he's busy, will always send the students, including PhD students who are registered in Germany, so postdocs, to Dr. Spectra, who's an African. Even another student of ours, when he demonstrated his skills in NMR, the question he has been asked is, how many NMR machines do you have in Nairobi? And they all assume there must be 3, 4, and high field. But when he told them we don't even have a single working NMR, I was surprised how he became an expert in NMR. So it means even in darkness, when we are forced, be able to see. And I think if you are trained to see in darkness, in the light, you can see much more clearly. So the examples I'll show you is how our students have acquired skills and managed to interpret and come up with complex structures. The first example is on family Leguminosae militia. This work was done by a guy, an Ethiopian, finished and went back to Ethiopia. Now is equivalent deputy vice chancellor. Solomon, the trainer, is still a senior lecturer. He's still a professor. And I think I'm sure you all agree Solomon deserves to be a full professor. But at least we need to have the right place and the right correct opportunity. Solomon feels the responsibility to be called professor. He's shying away. Prof. Kerum will encourage him. Feels to that isn't it? So it's a guy, then when he was a student, selected several compounds. Him, along with others from militia, were selected and characterized over 100 compounds. And just to demonstrate skills we imparted to the guy in characterizing compounds. So it's routine, after you isolate, generate physical data. Sure, until you are forced, when you are writing your thesis, is when most of them remember to measure melting point or even run UV, even mass. But when we present the other way around. So after generating this, of course, the most important analytical tool for a natural product chemist is NMR. We had access to 800 megahertz NMR machine through collaboration. And the guy inspected this compound through his experience, just looked at 3 sets of doublets in the area between 4.36 to 4.64. And from his database in the head, he knew the compound he has is a 12A hydroxyrotenoid. That was why students have to enrich the database by interpreting or looking at various spectra. Of course, from here, the protons in ring B and ring C, each having 2 coupling partners, they appear as double doublets, 3 sets of double doublets, and that is typical of 12A hydroxyrotenoid. And here signed as shown. Then, of course, once you identify the skeleton, then it's a matter of knowing what are the substituents and placing them. So the first thing we saw was ring A, based on how these compounds are naturally prepared. It's always substitution at carbon 2 and 3. And as expected in ring A, there's only room for 2 protons which are para-oriented, and we saw 2 singlets. So ring A is covered. Then the substituents here identified are methylene dioxide, that peak integrating for 2 protons. That kills the pattern in ring A. In ring D, you have 3 mutually coupled protons, and this is in agreement with a monosubstituted ring D, where from genetic background, that substituent must be at carbon 9. Then this pattern, doublet for this, and double doublet, shows the substitution part of the protons in ring D, which requires substituent at carbon 9. And that substituent is methoxy group. So from this, here's a proposal. Carbon allowed him to confirm by showing the sp2 hybridized carbon atoms and also the sp3 hybridized carbon atoms for ring B and C. He confirmed the connectivities through COSY. So that connectivity is confirmed through COSY, and the connectivity also in ring D, that pattern was also confirmed from COSY. Then from HMBC, you observed which proton is neighboring which carbon atom. So as shown in ring A, H1 is connected or related to C2 and C3. The same way, H4 is connected to this, so he confirmed the pattern in ring A, and then the methylene dioxide also position was confirmed. Then in ring D, then proton 8 correlates with C9, and H11 correlates with C9, so with that, the flat structure is confirmed. And also the methoxy position was confirmed through HMBC. This compound has 2 stereo centers. So even if the flat structure is as shown, Since it has two stereocenters, there are 4 possible stereoisomers. So of course, even in biological system, stereochemistry plays a big role. Enzymes chiral, so we have to also identify the absolute configuration of the molecule. So to do that, protenoids being all class of compound, there are empirical rules. In literature, empirical rule first allows the guy to determine the relative configuration. To determine the relative configuration, just looked at the chemical shift value of proton 1. So we have 2 relative configurations, cis and trans. When it is cis, the molecule will fold. When it falls, that proton will not be within the deshielding zone, will not be influenced by that carbon. So when it is cis, means it falls along that junction, the BC ring junction. Since it falls, typical NMR value is between 6.6 to 6.8. When it is trans, then the molecule will not fold, it will have almost a planar structure. When it has almost planar structure, despite the molecule being chiral, then this proton will be in close proximity to that carbonyl. So carbonyl will shift its position to high frequency or low field to high delta value. So that clearly is trans. If it is trans, then at least out of 4, he eliminated 2 possible stereoisomers, and his left is to decide whether it will be 6AR12AS or 6AS12AR. To decide that, he generated, these are the 2 possibilities. He generated CD or electronic CD and determined the specific rotation. So again, using the table in this proposed by Slade, then of course it was straightforward for him to decide which one. Positive caught an effect, 340, and negative at 300, and a high positive specific rotation fits with 6AR12AS configuration. And lucky for him, it looks a simple molecule, but it was new then. And of course, being a flat structure, he managed to get good crystals and he confirmed the structure with X-ray crystallography. So that way, he characterized several compounds. Some are isoflavones, protenoids, and all the red indicated here are new. So several compounds, even from other Miletia species. So I'm sure now, even if I don't show you how these are elucidated, you should trust me. It is only when we have complete structures, flat structure and absolute configuration known, that's when we go to the biology lab. Then for us, we look at the history of the plant, traditional use, and look for partners. So in biology department, my other African brother, Professor Kabaru, I think we have come a long way with him as well. Should I say over 20 years? Then he's in charge of the lab where he keeps mosquitoes. Just get the mosquito eggs, hatch them, then transfer it to Petri dishes. after they have attained a certain larval development, and then even a chemist can do some biological testing. So this, we're able to test our compounds. The first plant we worked on is Miletia dura. The seeds, as you can see, the larvicidal activity against Aedes aegypti is quite high. And then for us, we test all the compounds we have, whether they are isoflavones. In this case, isoflavones are not active, they are not insecticidal, we are not surprised. But we have two rotenoids, and well known for their insecticidal activity against other insect species. When we tested this, they are substantially active because they fulfill the structural requirement for a rotenoid to be active, need that chirality. As you can see, it must be cis-oriented. So is that V shape. This was active. We were not surprised and we were happy. And it shows quite potent larvicidal activity. Remember, that cis-B serine junction is a requirement for this molecule to fit in the enzyme surface and be active to disrupt the reduction oxidation process within the cells in the insects. We also had dihydrorotenoids, where you can see here there's no chirality. You can see that is a completely flat molecule. It's not flexible. Since it is not flexible, it could not enter into the enzyme pocket. This is well established, and this is not active. Having that flat structure where chirality is lost, because you can see at the junction, we have a double bond. So it's a flat, rigid system. And as expected, this is not active. We also isolated two compounds which are unique to this plant, Miletia dura. If you compare these structures, they are quite related in terms of structure to degulin and rotenone. Like degulin and rotenone, degulin and tetrasin, you can see they have the same configuration, cis configuration. The substituents, both of them, so they fulfill the structural requirement for insecticidal activity. Then, like degulin and tetrasin in ring A, these two are also oxygenated. But of course, oxygenation is not toxic. By then, the importance of substitution in ring A was not known. So the only structure requirement is that chirality wants to be. To our surprise, they are completely inactive. We have tested this repeatedly, they are inactive. So then simply means in degulin tetrasin, so we demonstrated that for the first time. Then we moved to advanced Jamycin, isoflavone is not active, and it's suppressed. Chalcons is also not expected to be active. Then, when we looked at our rotenoids, as you can see, the configuration here is trans. Remember, for rotenoids to be active, you need that V shape. But this is trans, nearly flat. So we didn't expect any of these rotenoids to be active. Say flat because in the crystal structure, you can see that is nearly flat because of that trans configuration. So because of that, we didn't expect any of this to be active. Moreover, we have demonstrated, as shown in the previous slide, it is metoxic groups which are important. The example we had in the previous slide, 2 compounds having methylene dioxide were inactive. Now, all the 5 compounds do not have cis configuration. They do not have metoxic groups. Instead, they have methylene dioxide. We didn't expect any of this to be active. Then, still, the crude extract is active. But when we test, 4 out of the 5 rotenoids were completely inactive. To our pleasant surprise, the 5th rotenoid, despite having trans p-serine junction and methylene dioxide, showed potent larvicidal activity. And of course, when something does not fit with the accepted knowledge, before you report, you have to be sure, and we did multiple testing, and the larvicidal activity is consistent. Then we say maybe this activity is unique to Aedes aegypti. So we had an opportunity access the Anopheles gambiae, the vector for malaria. Then this compound was also showed potent activity, even more potent than Aedes aegypti. At least by then we knew this is compound to follow further. So still, we have not tested to other insects against other insect species, and that's what we want to do. Then, of course, we know rotenone is cytotoxic. Then we tested Uzara rotenoid A for cytotoxicity both on cancerous cell, normal cells. It's completely inactive. But of course now, at least at this stage, we knew this compound is not toxic. So we said Uzara rotenoid A is the first trans 12-hydroxyrotenoid to show larvicidal activity. And no toxicity to human cells. Then, of course, now those who are keen on mechanism of action, this compound will be a good candidate. The second example I have for you this morning is on tephrosia. And again, I will not bore you to go through the structure elucidation, but to demonstrate how our students not only are capable of, remember, even without NMR machine at our disposal, not only they are capable of elucidating their own structures, as you'll see, they are even capable of criticizing published data. This story is Caroline, who just graduated with a PhD. So we also attract beautiful ladies in our group, not just handsome men like Caroline isolated compound. I'm sure now, trust me, she proposed this structure, including the absolute configuration through the method I demonstrated in the earlier slides. This class of compound are called beta-hydroxy dihydrochalcones. And of course, just to tell you how she did it in the proton NMR, you know it is beta-hydroxy dihydrochalcone, that pattern for alpha and beta, each of them appearing as a double doublet. Beta being oxygenated appears at low field, whereas the two alpha protons, if they are on the same carbon, since you have chirality in the molecule, they also couple with each other, each appearing as a double doublet. And then she did all the NMR analysis, CD, and including carbon, fits very well. So again, the focus is on the atoms shown in circle. So the carbonyl is clear, the beta and alpha carbon atoms also fits very well. And again, we sometimes undermine the value of mass spectrometry. There's a tendency of over-relying on NMR. As I will see later, it can be critical. So the mass also fits in terms of the protonated molecular ion and also the basic. So at least she had good set of data to support the correctness of a structure. Then of course, now she has two examples. And naturally, when she was drafting a paper, these are both new compounds, she checked literature, beta-hydroxy dihydrochalcones, it's a small class, there are few examples in literature. And compounds 5, 6, and 7 were published as recent as in 2021. And these compounds actually were proposed to have beta-hydroxy dihydrochalcone basic skeleton. Then she compared the NMR data for protons alpha and beta. Her data and the published data for compounds 5, 6, and 7. Specifically, for one of the protons at C alpha, her data is substantially different from the published data for compound 5, 6, and 7. You can see it is completely different. So here, either she has to go back to her data and find out what is the problem, or if she is sure about her proposed structure, scrutinize the literature data. Then, if you check here, beta-hydroxy dihydrochalcones, the beta carbon is oxygenated, like in flavanones, carbon 2 of flavanones is also oxygenated. Then the alpha protons, alpha carbon, similar to carbon 3 of flavanones. Then we have done a lot of flavonoids, including flavanone. So when we saw this pattern, and when we compared with these flavanones in the literature, as you can see, this allegedly proposed beta-hydroxy dihydrochalcone the values are more similar to a flavanone than the hydrochalcone. You can see that pattern. The green and blue you can see are comparing, and quite distinct from the red. The red is Caroline's data. Now, she also went ahead and compared the carbon chemical shift, again, for alpha and beta carbon and carbonyl, and also in the flavanones for carbon 2, 3, and 4. Again, you can see for this ring, for position 2, 3, and 4, the carbon chemical shift of carbon 5 compared to Caroline's compounds 1 and 3 are different. 6 is also different, similar to 5. 7 is different from 1 or 3, but similar to 5. And then rather, this data fits better to a flavanone skeleton than beta-hydroxy dihydrochalcone. You can see 5 is alleged beta-hydroxy. The A's represent the data for flavanones. You can see 5 and 5A are similar, 6 and 6A are similar, 7 and 7A are similar. Then we also did some calculations. It's a simple calculation Caroline did. Even the calculated values, ours fit with the calculated for beta-hydroxy dihydrochalcone, but for compound 5, 6, and 7, the data fits more to flavanone skeleton rather than the beta-hydroxy dihydrochalcone. Still, 5, 6, 7 were published as beta-hydroxy dihydrochalcones. The NMR, the pattern is similar, can be confusing, but the question we asked, there is a whole 18 atomic mass units difference between beta-hydroxy dihydrochalcone skeleton and flavanone. So this should have 18 atomic mass units above that of the flavanone with the same substitution pattern. That is a difference. If somebody has run the mass, could not miss. Remember, Caroline has run mass and it fits. When we check the literature, conveniently, some of them did not publish the mass data. That's been dishonest. And you see, they have already fixed their mind on beta-hydroxy dihydrochalcone skeleton. When the mass is not fitting, they just ignore it. Whereas for compound 7, we actually saw a peak at 289, around 289 M/Z value. So of course, when they saw 289 for compound 7, they interpreted this. The molecule must have lost water. Dehydration of alcohols is common. But that is overstepping. If you have not seen a peak for it, one can do the analysis, the measurement at low energy. But assuming the structure, this structure is correct, and accounting for that peak, which should have been molecular ion peak for a flavanone, they interpreted it as if that molecule has been dehydrated. Yes, dehydration occurs, but especially when the literature is confusing, you need to do a thorough work and generate all set of data to fit your proposed structure. From all this, you agree with me, these structures are wrong. And Caroline was convinced. She also convinced us. And as report to the new structures, we also revised these structures to flavonoids. And this was published only this year. So again, to demonstrate, even in darkness, our students can even see mistakes from European laboratories. So that also shows you we do our best to train our students. The final example I'll give you, similar, is on Asphodelaceae family. Again, we also have beautiful ladies. We were interested on Cnifophia, and Ethiopia is a center of diversity for Cnifophia within Eastern Africa. So I had an excuse to go back home to visit my mentor, Chronicle Garden. He allowed us to collect and carry the sample to Nairobi. And we isolated such diameric complex molecules. It's a long story, but why we needed such compounds in good amounts and also related compounds is because these compounds show very good antiplasmodial activity. Now, I did not have enough excuse to go back home. The NAPRECA activities used to sponsor us. And then said we should find, if not Cnifophia, a related plant from Kenya, which we are hopefully to produce such compounds. Like Cnifophia, bulbin is also, we have demonstrated, it produces compounds of this sort. So then, Nagera, students then, remember, his task was to isolate such compounds or related one complex structures and pursue the antiplasmodial assay, hopefully even at in vivo level. He isolated a compound. If you look at the NMR data, it doesn't look that complex. It can't be obviously complex structure of the ones the beautiful ladies have isolated. In a way, Nagara was disappointed. It's a small man, but the heart is big. So he said, how do I isolate a simple compound? Then it is related to those of you who have done quinones. The simplest and most common quinone is called chrysophanol. And he simply said, that my masters have isolated more complex compounds. At PhD, chrysophanol, it's like lupiol, you know, those noises for him. But he starts looking at it more critically. Chrysophanol, if you check, it has two hydroxyl protons, which could form strong intramolecular hydrogen bonding. So in our experience, when you have hydroxyl group paired to a carbonyl, you expect peaks above 10 ppm. He expected 2 of them, but he only saw 1. But OH are elusive. Those of you who have done NMR, sometimes you see them, sometimes you don't see them. But of course, even in the carbon, you expected 2 downfield shifted carbonyl groups above 180. You only observed 1, was expecting a peak here, it is not there. Instead, it has been in this spectrum, it appears at a high field, low frequency. Not 200 or 190, but around 169. But now, one way of confirming whether you have OH group or not, simply methylate. Yeah, from this, it's sure that he has one chelated or intramolecular hydrogen bonding. He's sure of this ring A, typically like chrysophenol. Ring C is also like chrysophenol. He has also seen this, but now here he has an issue. Doesn't see that carbon. Rather, it is upfield shifted. Then he compared the mass of chrysophenol with that of compound F. You can see there is 16 atomic mass units different. Now he said, means it's not carbonyl here, it must be a lactone type. If it is a lactone, obviously the carbonyl will shift to high field, then that he thinks is his compound. Then he went to HMBC. HMBC will only tell you story around protons, and especially when you are biased, even HMBC will tell you what you will show you what you want to see. But still, he was not satisfied. Still, the issue is why is only one OH visible? Why is the other one not visible? He methylated this. He said he obtained a dimethyl ether. He ran the proton and carbon, but notice the carbon, one of the carbon signal, to his dismay, is not the normal range for a methoxy, slightly upfield shifted. But then he has this working structure, and he looked in literature, and he saw there are examples like his 2 hydroxyl groups, and the lactone group are called seco anthraquinones. Some are even methylated. So he said he has a 7 new seco anthraquinone to report. He has even started drafting a paper. There's always a flip side of the story. Now start looking at the data in literature, and he's more critical. And these papers range from 2001 to about 2014. In all cases, they only report one hydroxy group. They have not reported the other. In all cases where there is reported a methyl ether, you can see consistently these methyl ethers, in some are around 52, 53, proper methoxys 56. He also compared the carbonyl shift. In all of them, including his, the data is within this range. We saw all this, all the evidence is pointing that his structure is correct. Until he came up with another paper. It's not a seco anthraquinone, rather here the carbonyl is reduced, so sort of hemiacetal. And this, they have reported 2 chelated or intramolecular hydrogen bonding. They have also reported the carbonyl at 197. Remember, everybody else is reporting slightly lower value. If there are 2 hydrogen bonding, such carbonyl should come close to 200. There's no way such carbonyl can appear in that proximity. And this was published in Journal of Natural Products. At least the referring system there, it's more strict. And of course now he has to do further research. So he put a question mark now. That paper was kept on hold. Then, courtesy of DAD, he has a privilege to visit Bielefeld University in Germany. And this is the one I told you, Nagara is Dr. Spectra. So he was playing a professor role when he was in Germany. So now he approached his professor because the respect he has gathered to allow him run this for X-ray. I'm sure the prof will not spare valuable X-ray time on such simple structures. But Nagara convinced the professor, and he was given time. The instrument he ran X-ray. To his surprise, X-ray showed it is not secoanthraquinone, it is a xanthone. So NMR is useful, but that bias can make you make wrong conclusions. Ultimately, if you have a chance, X-ray is quite useful. For us, X-ray is out. Only thing we do, we are still working in the darkness without NMR. But again, X-ray, we start looking back to the NMR data, including the methylation he has done. That methoxy 56 is correct. I'm sorry, it's not visible. This is about 53. 53 is not for normal methoxy. Those of you have done with carboxylic acid derivatives, methyl esters appear around 53. Now it's clear. Even the NMR evidence points to a xanthone, and that is the correct structure. So his working structure, lucky enough for him and for our repetition as well, we did not publish this. Then of course, now we have sharp eyes, since we have trained our eyes to see in the dark without NMR, bold enough to revise all the literature of secoanthraquinones. They are all xanthones. Yes, this was also accepted. When you revise a paper and it is submitted from the dark continent, it's not easy to be accepted. But at least our kerosene lamp technology has been respected enough, it was accepted. Then of course, when you do NMR on natural products, proposing a structure, no matter how good you are in the spectroscopy, should always justify its biogenetic origin. Even if it is speculation, you have to do it. So when we check, xanthones and anthraquinones coexist. They coexist, and xanthones of this sort can be, and it is demonstrated that they can be derived from anthraquinones, cleavage, rotation, then cyclization will convert anthraquinone to xanthones. So the plant, we already know, contains anthraquinones such as chrysophanol. So the co-occurrence of xanthones genetically is acceptable. But the message here is you all practice this. You propose a structure, compare it with literature. If our data fits published data, hooray, we rush and publish. So usually jump into bandwagon. So the message here is make sure all the data without exception point to the same conclusion. It's only then you should publish. Then can also criticize and correct literature. I think Solomon, thank you. That is my part. The second part was supposed to be workshop, isn't it? Yes, yes. Yeah. For now, so thank you very much.
Everything is quiet. Or is it my computer?
It is also quiet here.
Can I say something on behalf of what
Same
Prof.
with
has just presented? Yeah.
I can sit.
Together with it. Find some space. Okay, morning, Assalamu alaikum. Solomon wanted me to introduce you to the center in Karachi called International Center for Chemical and Biological Sciences. The reason why he wanted me to do this because of a very simple fact that ours is a developing country as well. Pakistan is a developing country as much as Kenya or other developing countries in Africa. And over the years, we were able to develop a good quality institution. Now, the intention is to tell you that, institutions are not developed in vacuum. They have a context, and there is always certain important factors which play an important role in the success and failures of institutions. We see lots of examples of very successful institutions, and certainly at the same time, we see examples of failed institutions. Now, I want to give you the example of an institution which is reasonably successful. Of course, there is never an institution or no institution in the world which claim to be the perfect one. We have our own problems. And as we will proceed, I'm going to tell you our problems also. But this is an institute which was established in 1966. So it's a legacy institution, roughly about 56 years of life. It's also a UNESCO center. And okay, so I'm sure that you'll not be able to read this. There is no intention for me for you to read. I just wanted to show you the genesis of an institution which established in 1966 in a small laboratory. The 1/4 of this room are 3 phenomenal people from 3 different backgrounds. One from biochemistry, the other was very high quality natural product chemist. The third one was a modern chemist who came from Cambridge. All 3 of them, 2 young men and 1 very old man. And when I say very old man, because
He was already 77-year-old and he was establishing a new center. You know, after being chairman of Indian Council of Scientific Industrial Research, retiring from there, establishing Pakistan Council of Scientific Industrial Research, retiring from there, becoming Vice Chancellor of University of Karachi, retiring from there, he come afresh to establish an institution, and he wanted to help the young people, Professor Taur Rahman and Professor Waqaruddin Ahmad. So he gathered these people in a small laboratory, and then from that very day on, he continued to serve this institution until 1994. So he remained the patron and director of the center up to the age of 97 years. And his last paper was published 4 months after his demise. So you can think that, you know, he was, I'm going to show you the picture. So from that very point on, this institution is now a huge institution by all definition. You know, 17 research centers spread over 200 acres. There's a huge campus. And no undergraduate program, 100% graduate program. So only extremely proud. You can see this person with white hairs. He was a legendary professor, Salim Usman Siddiqui, who has done work on Ravelfia serpentina. He was the one who isolated many companies. He never believed on patents. He was against patenting. And he said anything which we discover is the common human intellect and property. And then Professor Taur Rahman, who's a legendary scientist who came from Cambridge. Professor Siddiqui was the founding member of Vatican Society of Science, Fellow of our Society. So all these big people together with me, a young man. I was a young man. Can you believe it? So together we all have worked, and you can see some faces which are not scientists. These are the people with great hearts. So they brought millions of dollars from their own savings and invested into the institute. And these are completely private people with no scientific background, but the vision to invest in the future of the country. Now, as a result of it, we have an institute where we have two big sections, one for chemistry and other for biology. And the philosophy is that the most exciting things that are happening at the interface of chemistry and biology. Professor Bay has presented his work on, dengue, lipocytal activities, fascinating chemistry, absolutely classical, high quality chemistry. So you combine different disciplines together, and then you are able to do lots of, because you remember that problems, whether a disease or environmental problem, they are truly interdisciplinary, and people from different disciplines have to work together. So this institution is a very unique place where people from absolutely different backgrounds, from genetics to nanotechnology to biotechnology, from organic chemistry to pharmacology, they all work together under one roof. And this is some of the chemistry centers. These are huge buildings. And each building has a very unique feature. It has its own equipment, its own team, its own faculty members. Sometimes it's their own budget. So these are Chemistry Center, HEJ Institute of Chemistry on your right. And then there is Professor Tarman Laboratories, there is a nanotechnology center, biotechnology center, because we work with medicinal plants. Professor Wolfgang Felter of Folter Center, there was a German scientist who remained with us, is a truly international center because we had many German, Japanese worked for years and years in this institution. So there are so many of these centers in the field, broad field of chemistry, nanomaterial, biotechnology of medicinal plants, organic synthesis, textile chemistry, medicinal chemistry, and others. And these are some of the biological centers, very high quality, molecular medicine center, genomic center, where we do lots of genomics, forensic DNA, serology. Then there is the National Institute of Virology with BSL-2 and BSL-3, large BSL-3. Then we have our own animal houses in which we are capable of designing our own animals. And when I say, it means that we can produce disease animals by ourselves, either by inducing the disease or designing them right from the beginning by using gene knockout, gene knocking technology. And then we have a full-fledged clinical research center, which is a DRAP, Drug Regulatory Authority approved center, WHO designed center for clinical research, phase 1, phase 2, phase 3 clinical trials, which we can conduct. Okay, so what makes our institution different from the others? We are one of many centers. Pakistan has 159 research centers, and we are one of them. We are exactly the same people as the other. What makes us different? Number one, right from the beginning, we decided to have contractual appointment of faculty. No tenure to anyone. If you want to stay here, get good salary, good facilities available, but will not make you permanent. We'll evaluate you every year, and then every 3 years, and then every 6 years, then every 9 years. And if you are good, you will stay with us. If you're not good, you'll go out. So this exit path was always there. This contractual appointment was very, very important, including director. Annual internal evaluation. Every one of us, my colleagues are here, every one of us are evaluated every year against key performance indicators. There are 1,000 marks, 700 for research, 300 for teaching, 600 for research, 300 for teaching, and 100 for services. Everything is quantitative. You can measure by yourself, self-assessment, and only this 100 marks, which I give with honesty, the rest of it, everything is so quantitative that you would know exactly what is your teaching score, what is your research paper score, what is your patent score, what is your citation scores, how much the score in terms of bringing money from outside. Everything is completely quantified in terms of key performance indicators. And then institution itself is evaluated by the top people. So we have a 5 years of, every 5 years we get the team of scientists coming from the World Academy of Sciences, sponsored by the World Bank, and they come and evaluate the center. And right from the day one, we told them, you don't need to praise us because we know that we have lots of problems. So tell us our problems. So they give us a long list of how bad we are. Your safety standards are very bad, your security standards, your students are taking lots of time, all bad thing about us. A whole list of, and whenever I see this list, I feel as if we are the worst people in the world. But then we work for 5 years to improve it. And then they come again and they give us few points and they assess our success and failures. So key performance indicator, as I said, that how many PhDs you have produced, research funding. And then we have a very, very different paradigm. It's called reverse pyramid, where young people. So professors are
funny X-rays are cure
while young people need to be supported. That is a very different kind of So this is the kind of assessment we have every 5 years. The last one was in 2019. We are preparing for 2023 and 2024. And these are people from 98 different countries, government, academics, research standards, infrastructure, instrumentation, spectroscopy, safety, chemical safety, biological safety. So there are several other characteristics in classes. You know, we have invested about $50 million in our instrumental facility. And if we don't have good quality engineers, they're completely fail. They are people with the golden hands. They're quite trained, nurtured, retained, and respected. So the engineering staff, the senior most engineers in the system is in the same grade as the director general officer. So we have, you know, over a dozen engineering staff capable of maintaining NMRs and mass spec and other complex machines. And then open for accountability. We have been all the time under review of different kinds of, you know, from international, national. So we've been always, and then another important thing, we spend 70% of our budget on research. You know, this was right from day one. Professor Siddiqui said, I've said, you know, he made very interesting metrics because he actually worked in 2 different countries, studies in a lot of different countries. He said institutional threat. What are the factors which eventually lead to a complete collapse of the institution? And one of the institutional threats which he has identified was that you over-employ people and then you have nothing but to pay salaries and no money for research. So by statute of the institution, we cannot spend more than 30% of our budget on salaries and scholarships. The rest of it has always been, should always be available for library, for spectroscopy, for research. So every research faculty, every faculty member supposed to receive, every faculty members receive funding from the institute, and is also matching grant, which we have recently planned to resume it. If you bring $1, we'll give you $1. If you bring $0, you get $0. And so this is how we, and then, you know, we have a very large doctoral program. We, this is also very unique that every student receives scholarship. Everyone receives scholarship because there was a strong belief, Dr. Siddiqui used to believe that students come at the age of 23, 24, and if you do not only provide them required financial support, they will not be able to focus their work on their work, and they would perhaps be spending time on doing different jobs and all that. So every one of them is a recipient of research fellowship, each one of them, and all the time. So this is how, and then every student is co-supervised by leading people like Professor B. So, you know, every student has a foreign co-supervisor. This is also one of the important factors of our. So we good in synthesis, chemistry and biology. My colleagues run molecular biology laboratories. They can synthesize proteins of different types. We are good in analytical and diagnostics. We can do analytical work and diagnostic, genetic, cellular, and chemical levels. We have excellent instrumentation facilities, and we work towards training people in biological, chemical, and clinical sciences. So we have done lots of things. I just want to show you that the first human genome. So we do now human genomics, complete human genome. It's very, very routine. You can go and have your genome done in the institute. We have done total synthesis of fit plasma was synthesized for the first time in our institute. And we have a drug called ISOX, which is a phase 2 clinical trials, anti-epileptic drug, anti-cancer dietary supplement, which I have presented, anti-Leishmanian drug, which is already in clinical use. We have done lots of genomics disease, deglaria, dengue viruses, lots of different things also, and then many other things. We are very accredited facility, so entire institution, literally hundreds of these laboratories, each lab is accredited with 9000-17025 and 4001. So these are ISO internationally accredited facilities and many other accreditation. We have good quality infrastructure. For instance, our NMR facilities facility is among the best in NAPRECA. We have 13 superconductors, 300, and there are many of them are equipped with cryoprobes and dual detectors and another thing. Then we have a very high quality virus facility. So we have 5,600 square feet of BSL-3. Then we have several BSL-2 laboratories, several dozens BSL-2 labs. And, we have, this is very important, we have banks, we appreciate banks are important, but we have banks not for money, for molecules, for cell lines, for microorganisms, for viruses, for other things. So these banks they're bank managers sitting like me, with big freezers behind, and you can borrow molecule, or you can give your molecule to them, and thus you share, and you can go and you can borrow. So this is BSL-2 and BSL-3 facilities, a lot of them, just few photographs. And we have done lots of work during COVID-19. We were the largest research center with which we have done, which PCR test. We were the Center for Genomic Surveillance. We have studied vertical transfer for the first time in the world, mother to baby transfer of. Then we have conducted several clinical trials also. And then we are public service organization. We generate lots of funds by providing services to the industries and to the government. We support over 600 industries. We have lots of industrial internships and others. This is our NMR park. So we have been the oldest users among the oldest users of NMR in this part of the world. So we started using NMR in 71 when we had this permanent magnet NMR. And the first superconducting NMR was procured in 1980, and that still works. And the manufacturing number of that machine is 101. So this 100, before us, only 100 NMRs were sold, and that 101 machine, the oldest functional NMR machine, is in our institute, oldest functional superconductor, and it works for decades. And it's because of, I think, the quality of engineering staff, and of course we have more than 800 megahertz. And this is a cryoprobe. So it's like its resolution is about 1 gigabyte machine, and it has 2 detectors. So it can detect proton carbon at the same time, proton carbon, and then can also detect nitrogen at the same time. Also, it can detect several nuclei at the same time. And then of course we have several of these X-rays. Our mass spectrometry facility is also very good. We have lots of imaging. We are the best imaging center in the country. We can image animals, we can image microorganisms, others. So we have lots of these imaging facilities. These are, I don't know what these are. These are also microscopic. We do single cell physiology. So this is a very unique strength of our institute, that we can do from bacteria to single cell. We can get a single cell from the brain and we can do physiology of it. So We do lots of this electrophysiology. Then our genome is, we run many second generation genomes. And there were a national facility for laboratory animal research where we have several genetically modified animals. And of course, we run the best material testing laboratories. So from scanning electron microscopes to transmission, electron microscope, AFM and others. And so a lot of these facilities that we have a central computing system with very good quality libraries. In chemistry, we have a literature from 1739. So the complete literature from 1739 till today is there. And of course, lots of biological, there are many libraries, digital. We also connected to Pakistan satellite number 2. So we are the digital education center of the country. We liquefy our own nitrogen and occasionally we liquefy our own helium. So we have the capacity of recovering helium and liquefying them. It is a completely solarizing institution. So completely off the grid. No external electricity during the daytime. Is a 2000 kVA solar panels and in-house hotel facilities, college. We have been internationally recognized by lots of prizes. Lazata has received UNESCO prize, Chinese prize. I am the recipient of Mustafa, peace be upon him, prize, which is considered to be a Nobel Prize of the Muslim world. We also have international linkages, and we're looking forward to have very good collaboration with your universities. So we have been able to
Yes, welcome back. So can we have a Q&A session? Do you have any question for Professor Iqbal? And probably you can unmute and ask a question, or you can type your questions in the chat. But in the meantime, any one of you in the audience, it is possible you might have some question. You can go ahead and ask your questions. Anyone with a question? Yes, Geoffrey. It's better you come closer so that the you can use it. You can use it from the other side. Yeah, so that's easier. Huh? Yeah. Mine is not let's see. So the other thing, I want to ask, because we also have our team for the science and technology here in our ministry. And I don't know how it's possible for the center to be run in a very autonomous manner. This is the thing I was saying. From University of Canada website. Really like, so we want to learn from you very good model. Okay, as Solomon mentioned, if you give us in speech economy with a conflict, you cannot have only economy and that you will do whatever you want. There should be an economy. So this is which is a very strong quality.
They made up convinced that if you want us to be successful, then you know the government, you should be able to hire people. You should be able to attract funding from international sources, and sources. You probably will come. So you are not running other countries for this stuff, there are countries for unless they haven't leveraged from that. I think this is the model. Lots of mistakes. Of course, we have we have been so if you solve this year, you find this institution, if you come next year, you find this it has been growing every day, every day. You have seen a picture of ex-president of Mauritius. She's our alumni, and when she assumed power as the president of Mauritius, she decided to come to Pakistan. This was her first visit, and her first stopover to our institute. So this has been, our alumni all over the world are one of our greatest strength, and they are literally everywhere, literally everywhere.
Thank you very much.
Yes, sir. Well, Professor Chaudhary, I have the pleasure, of course, to be one of your students. It's my great pleasure. And also, I had the pleasure to attend the 15th International Conference in Natural Product Chemistry this year in ICCPS. I'd like to ask about, I have witnessed actually the great young scientists, master and PhD students, their excellent scientific background. I'd like to ask about what strategy do you prepare these young scientists for the future? Thank you.
Okay, so very important questions, because all 600 of them, there is absolutely 100% merit based. So there is no quota, 60–70% of our student body are these young girls. So based on the merit from anywhere in the world, you can get the admission, and you will get the full support throughout your program. And then once you select people on the basis of merit, then we have a very extensive coursework in which we provide them excellent opportunities of learning from the best of people in the disciplines, and also possibly the opportunity of going abroad for a certain period of time and coming back. So there are numerous schemes through which they can go abroad and come back and learn. And there are so many things. Yes, for example, every student has to have a GRE, graduate record examination, passed. They have 48 credit hours of courses. They need to deliver open public seminars. They need to write a research proposal which is fundable. Something which supervisors eventually submit and get the money on. And then right at the end, they all have to something very interesting. For chemistry and biology, they cannot really go out of the institution unless they have personal viva conducted by us. It's not a formal viva examination, or it's by us. So we make sure that only the best come out. So that's how we were able to, because I think without high quality graduates coming out and their employability, the institution would perhaps not survive.
I think, thank you very much.
Thank you. Thank you. I think there's no need. We can resume our work.
Were there online questions?
Yeah.
Were there online questions?
Let me see the no.
Maybe in the chat.
So there's a question, an online question on the line from one Anderson from
Okay, I can tell you some secrets which I never told you. For instance, the director of the center and our center is much larger than universities, I can tell you. It still use a car which was purchased in 2007. We don't change cars. We don't have any personal staff. We don't have offices. We all sit in our own laboratory. All of us, including Professor Atta, Dr. Siddiqui. Our administrative cost on us is very, very little. So we save money, We don't spend money on ourselves. We organize international conferences. We don't spend a single penny of the institution. We raise funds for a different thing also. We have been always looking for money because without funds you can't. And there's so much of opportunities. Of course, one needs to look into opportunities nationally, internationally. But it's important once you purchase these equipment, even if it is a small equipment, you should be able to run. When we were establishing this institution, like any developing country, we had this problem of electricity. We didn't have proper electricity backup. And if you are running NMR on external electricity, it will be gone in a daytime. So we created 3.5 megawatt our own electricity generation system. We had our own powerhouse with UPS and everything, because without these backup, you can't do that also. So my response to your question is that, of course, these things are getting better also, but we have lots of challenges because number one, that these equipments, you need to constantly replace them. And for that, you need to look for funding. So we keep applying for funding. We spend a lot of time. The director's work, my work is largely of raising funds. This is my first priority, because if I don't do that, the institution would not. And there is also one phrase which I would like to quote from Dr. Siddiqui's life: the science institution either go up or go down. There is never a plateau in their life, So either you go up or you go down. And I think this is the philosophy behind it.
Thank you very much, Prof, for opening our eyes and on what is possible, that if we dream and we work at it, we can all achieve. Such kind of, I would not be excited if it was in Europe, such a facility. If it was in America, okay, they can throw money at it. But all these things came through determination, hard work, and showing all of us, Ugandans, Kenyans, Cameroonians, Egyptians, who else, that is possible, Rwandese, it is possible to have such centers of excellence in our countries. The only thing that will stop you from achieving is that just because you say it's not possible. Ibrahim, okay? When you go to Nigeria, we'll want to see such a center established. Don't say it's not possible. It is possible. Okay, thank you very much, Prof. So we'll break for lunch and we'll resume our activities in the afternoon. Thank you very much. Okay. So what we'll do is to avoid traffic. What we are going to do is we will walk across the tunnel to the other side, and buses will wait for us. So we'll avoid all this traffic. It will take, I don't know how many minutes. We'll just do that, and then we through the campus, and then through the campus 5 minutes at Chiromo Club. So personal things, of course you have, if you have any personal things, please go with them. The rest of the things are secured. Yeah. Thank you. Yes. Okay. Yes, yes, yes.
We can walk to the car
Yes, yes.
and we can ask the driver to come here.
Yes, yes, yes. In fact, then we work.
Yeah, you want to go?
Yes.
Check it in. Of course, the tunnel, we were trying to do the tunnel. It's not come very fast. Was taking a deep.
You will read something or I should just start?
No, no, no. Yes, Professor Kerubo, I hope you are ready as the session of the chair. I mean, as the chair of the session, please start us off. Welcome everybody from the lunch. Professor Kerubo, you are muted. No.
Okay.
Yeah.
Thank you very much,
Hello?
Solomon. Yes, you cannot hear me? Solomon,
We are not hearing you.
how come? And I'm talking.
She's not being heard from the other side.
Hear me?
Is he ready?
Yeah.
But sorry.
Hello.
Oh, she's there.
So
Talk.
Okay, we can't hear you.
we are not hearing anything you are saying.
Oh. Try again.
No, it's me. I don't do touch.
To my end. It is your
No,
end.
we are not hearing you.
How come? Solomon? Can you find
Yeah, I
out?
mean, she's unmuted.
I'm unmuted and you cannot hear
Professor
me.
Kerubo, we are not hearing anything you are saying. I don't know if your gadget is allowing you or we have a problem from our side.
To your side.
Can you check can you go to the speaker, please?
She says she wants to start.
I think I commented on that.
Yeah. Okay.
Solomon.
Yes, now we can hear you. Start.
Okay, thank you very much. So members, welcome back again from the warm lunch. So I'm going to take you through the presenter's biodata, a brief one. So we have, that is Professor Dr. Atia Wahab. It comes from the organization which is known as Dr. Panjwani Center of Molecular Center and Drug Research, International Center for Chemical and Biological Sciences, University of Karachi, Pakistan. Professor Wahab obtained her PhD degree in organic chemistry from HEJ Research Institute of Chemistry, University of Karachi in 2007. She got her postdoctorate training from the Scripps Research Institute, La Jolla, USA, in the field of biology in the laboratory of the Nobel laureate, Professor Kurt Whatrich. She has completed her second postdoc in structural biology under the supervision of Professor Dr. Schwalb from Johann Wolfgang University, Frankfurt. She has established the first structural biology laboratory at Pakistan of the Dr. Panjwani Center of Molecular Medicine and Drug Research, ICCBS. She has more than 80 research publications in reputable journals in several fields of organic chemistry and structural biology, culminating to an H index of about 275. Important to note is that she has been granted with a number of international and national patents. Professor Wahab has won several external research grants and also supervised up to around 19 MPhil and PhD students. She is also mentoring a number of young faculty members and has co-authored 3 books in the field of organic and medicinal chemistry. She has received 4 awards and scholarships, including the Merit Scholarships for PhD Studies, the PAC US PhD Partial Support Program, the IUPAC Young Scientists Award, and the Productive Scientists Award. Professor Wahab has trained 72 Pakistani researchers and students from other universities and institutions in the field of structural biology and bioorganic chemistry, and also supervised and trained 37 foreign scholars in the field of bioorganic chemistry. The topic of her presentation today is biomolecular NMR spectroscopy and its application. Welcome, Professor Wahab.
Thank you so much for a very kind introduction.
Take this mic closer to you.
Okay.
Yes.
Can I sit here? Thank you. Good afternoon and Assalamu alaikum, everyone. So I'm very much honored and thankful to NAPRECA and comstech for inviting me to beautiful Nairobi and present and share some of my research. I'm very much thankful to Professor Solomon. His hospitality is just amazing. You know, he just we are just feeling like we are at home. We are not feeling we just come here for the first time. And I would say that people of Nairobi are very welcoming and very warm-hearted, and I'm so happy and delighted to be here. So I'm going to share some of my research work on biomolecular NMR spectroscopy. And first, I would like to say a few words about my journey. I started my research immediately after completing my master's in PhD. And then these Professor Daur Rahman and Professor Iqbal Chaudhry were my supervisor, and I'm so lucky that whatever I have achieved in my life so far is because of the mentorship of the great person sitting here, Professor Iqbal Chaudhry. He actually holds my hand and, you know, not only training, but to guide from very beginning in research and how to communicate, how to present, and how to defend projects from each and everything, you know, analytical skill for as a researcher. So I'm so honored, I'm so much thankful to sir. He's my mentor, not only during PhD, but rest of my research career in my life. Thank you, sir. And then I got postdoctoral training in Professor Woodridge. Actually, I did my PhD in chemistry, hardcore chemistry, phytochemistry, and synthetic chemistry. But then I worked with Professor Kurt Woodridge in Scripps Research Institute, and I learned structural biology from his lab, the hardcore structural biology, which was very different. And I would share my experience. It was not an easy journey for me. And I was always like complaining to my supervisor, Professor Iqbal, why you have chosen me to send here. Honestly, you know, it was so difficult at the time, but I'm happy that after returning back to my home country, I established the first structural biology lab. And I also did my postdoc with Professor Harald Schwabe in Goethe University, Germany. And from where I also learned many aspects of structural biology. And I would like to share that I'm not involved only in my own research, you know, because of this experience that I gathered over the year with my mentor, Professor Iqbal. Our group has been working in different aspects. So we have been working in biotransformation, as Professor Iqbal has mentioned in his lecture, and the pure phytochemistry from isolating secondary metabolites from plants and characterize them. And medicinal chemistry, you know, our teacher, Professor Iqbal always says that chemists should know how to modify the structure. So all the students who are working in our group not only have to, we expect and we train them that they not only go to the isolation, characterization using all spectroscopy technique, but they can modify the structure using synthesis. So we've been doing medicine chemistry from the compounds they have isolated or the drugs which are available to modify into different structure. And molecular and structural biology, this is my like my subject in which I've been working since my appointment. And then the other thing is aromatics, you know, different type of bioassays that our group has been doing using inhibition of different important enzyme. We have a molecular bank which about 19 or 20,000 compounds fully characterized that been isolated and synthesized in our institute. And then we are identifying, these potential so find some inhibitors of enzymes that are important for disease conditions. For example, aromatase, urease, acetylbutyrylcholinesterase, alpha-glucosidase, tyrosinase, anti-glycation, etc. And recently, we just started working on inhibition of A-beta 42 aggregation, which is important in Alzheimer's disease. And our group have been working in Parkinson's disease management and treatment. Dr. Iqbal also mentioned about detail in his presentation. And we have also been given the task from the government of Sindh, which is our province, to document and survey and document the medicinal plants that are being used by the people, because due to urbanization and access to the modern medicine, these folk knowledge that has been acquired by generation to generation over 1,000 years of time is being diminishing. So Dr. Iqbal's proposal was we need to document, and the government has approved, and we have we have actually report after survey of all villages, we have made a document, 2 documents for infectious skin diseases and women-specific diseases treatment. And we have been working on different type of cellular and in vivo model on bone fragility and biofilm inhibition studies. So these are the some key areas in which the group of Professor Iqbal Chaudhary has been working, and I'm privileged to work under his kind supervision. So now I'm going to start some of my research expertise, which is biomolecular NMR spectroscopy. Protein are those molecules which perform all the functions. So I would say in the drama of life at the molecular level, proteins are the one who actually plays the action. And you can think about that only a very small number of proteins structure has been reported in protein data bank. So it is estimated about 2 to 4 million of protein per cubic micron of bacteria, yeast, and mammalian cells, but only few of them are in the structure we actually be able to acquire. And we know that only 20 amino acids which are naturally occurring. So the rearrangement of these 20 amino acids that makes a big difference, and people are still struggling to identify, because once we have the structure of the protein, then we can design some drug leads that can control or inhibit, because most of the time disease conditions are relevant to some expression of proteins. So that is why the identification of these proteins, the characterization and structure determination are very, very important. And so we using NMR spectroscopy and other techniques, we can identify and study the protein interaction or protein ligand interaction. We can identify the arrangement of catalytic amino acids in the catalytic side and how they can interact with small molecules. And so it's very important that it's about more than 400 times sequences of proteins are being reported, but the structure are very limited. And there are various techniques that have been used to determine the structure of protein, which include NMR spectroscopy, X-ray crystallography, and nowadays cryo-electron microscopy, cryo-EM. So most of the time and if you see protein data bank, so most of the structure is being reported by X-ray crystallography, about 85%. And about 12 to 13% from NMR spectroscopy, and the rest the complementary cryo-EM is being used to visualize the big and aggregates of the molecule. But NMR has an advantage over other techniques, because in NMR, you can use the condition which is very close to your physiological conditions. So you can see the flexible part and how the protein would be present in the physiological condition. And NMR, as we all know, that it's a very versatile tool, and it can be used for ligand binding studies, dynamic studies, metabolic profiling, drug lead optimization, and structure determination. So the main purpose of my work is to determine the structure of the protein and its function, and annotate its role, its function. And we are also working on identification of using NMR spectroscopy as a tool to identify small molecules or compounds that can interact with proteins. And we can use it because this is very different from in silico studies, because in person, we have been like lots of things have been developing, but I feel like something that you really see in NMR, it's something real is happening. But in silico studies, no matter whatever you give the computer, they will dock with the molecule. So even there, if you do a real experiment, it's possible to get the real interaction or not. So my presentation is being divided in two major parts. One is the structure of protein determination, and second is the interaction of a small molecule with protein. So protein structure determination, it's divided into two parts. One, it's called molecular biology part, which include protein selection, gene engineering, protein expression, isotopic labeling, purification of the protein, optimization of the buffer, because proteins, as we know, that are very sensitive for environment and the pH change. So we need to optimize procedures. So we use basically it's multiple steps, and we use 3D NMR spectroscopy actually to get the structure of protein. And but the primary information that if the protein, we can use this protein for the structure determination using NMR spectroscopy or not, is being determined by simple proton NMR and 2D HSQC experiments. These are the two basic NMR experiments that we use to identify if we can take this protein further or not. So once we have the NMR sample, protein NMR sample in hand, we check proton NMR, and I would show you here. So looking this is spectra, these are, we can predict something. So if we have a folded and globular protein, so we have very dispersed spectra. You can see there are lots of signal. There are some signal which is below zero as well. There are some very dispersed, because NMR, proton NMR scale is just too small. It's just from, -1, -2 to 11, 12, 13 ppm. And that's a very small scale. And we have all protons here. And if you have a small protein, like about 100 kilodalton protein, so, 10 kilodalton protein, if even then it has the number of protons is like amazing, because in one amino acid we could have like several hundred of protons there. So these, we have all information there, But our problem, we cannot get and read the information from proton NMR. That is why we use 2D and 3D NMR to extract this information for us that we can interpret. Otherwise, all information is always present in proton NMR. So this is the basic experiment that have all the information. And if the protein is non-globular, and so it means like flexible, so you can see very sharp signals, and there is no signal below 0. So which shows if you compare these two spectrum A with B, then you can see it's quite visible that protein is folded or not folded. And in case if your protein is aggregated or oligomerized, so you could see this kind of spectrum. So proton NMR is very important spectra, which give the basic information. It has all information, but so far we cannot read all information about protein using NMR, proton NMR spectra. And then the second important technique is HSQC, which is 15N HSQC. 15N HS, as a chemist, we always use 13C HSQC, and here it's a carbon scale, but here we have nitrogen scale. So since we label protein, the problem here arises that we have, you know, and as NMR spectroscopy, we know as a chemist that the sensitivity of carbon and NMR and nitrogen is very low as compared to proton. We are lucky that we have proton that is very, very good and sensitive nuclei, but carbon-13 nuclei as abundance and magnetic ratio make it very insensitive, and similar is the case for 15N, which is NMR active nuclei. And so we have to label this protein with all 13C and all 15N so that we can get good spectra. Otherwise, in natural abundance, it's difficult to acquire good quality spectra that can lead for the structure determination. So if the protein is very well folded, you see, these are amide protons which are attached to nitrogen. You know, each amino acid is connected to the other with the amide bond. So there is NH, and NH is there for each amino acid is connecting. So this is some kind of basic tool. So it means if the protein is well folded, you know what happens when the protein is folded, then since NMR is very sensitive technique at the atomic level, so which makes that each atom is being either shielded, deshielded, or it has lots of different electronic environment. And this is the only technique in which we can read each single atoms. So, and if you have a protein like this, which here you can see that they are very separated molecules, amino acids, but there are this kind of overlapped peak, which shows that some part of the protein is very well folded and some part is flexible. So this may happen with the protein. So, and we can, this is the basic information which we get with 1D and 2D NMR. And when we have good quality spectra like this, then we decide, okay, now we have to move further and record the 3D spectra. And so I would like to now take you after this introduction to some of the protein structure that we have worked. And it's a quite tedious task. It requires several months of efforts, and lots of spectrum need to be recorded. So this is the one protein, which is DNA mismatch repair protein, mu2. And this protein is related to phosphohydrolases protein. And the main function of this protein is it actually kind of help in proofreading. So it's important that we should get the structure of this protein. So these are the amino acid sequence. It's 130 amino acids. And you can see these are the minimum number of NMR experiments that we have recorded. And each one, for your learning, I would like to explain. Since these are all inverse NMR experiments, so magnetization is being transferred from proton to nitrogen, then carbon alpha, then carbon beta. So this HNCA means, CACB means this from a magnetization started from proton, then nitrogen, then C alpha, C beta of the same residue and the previous residue. So it's very important experiment, and similar is HNCA, it goes up to the C alpha proton, and this is CO, means it goes to the previous carbonyl carbon. And then we have Toxis experiments, since each amino acid has side chain into it. So we need to have hydrogen toxi and carbon toxi to record all, to get the information of all side chains. So for this, we recorded for carbon toxi, CCCONH, and for hydrogen toxi, HCCCONH. These are all 3D spectrum. And then a very important thing, once we have backbone of the protein, we assign backbone means at least we should have like hydrogen, which is attached to nitrogen, carbon alpha, carbon beta, carbonyl carbon. These are the backbones which each amino acids are joined together. And definitely then afterwards work on the side chain. And when we have all assignments done, but we have to use different scripts to get this structure calculated for using the NMR plus NOSES peak. So these NOSES, that is the 3D NOSES experiments, like 15 N-resolved NOSES, C13 aliphatic resolved NOSE, and aromatic resolved NOSES. We get that we have all these spectrum. For each peak, we calculate, you can think we have a small spectrum, small molecule, we have the NOSES, it's very complicated. In a protein in which you can see the NOSES, intramolecular NOSE and intermolecular NOSE. So these are NOSES within the residue, intra-residue, and inter-residues. So we have to get these NOSE information, and the width of the NOSES peak, and this is the critical thing for which different programs scripts being used to calculate the structure using NOSES and our assignments. So here, once you have all done, so this is again HSQC, but now you know that each peak we already assigned. Okay, this is leucine 19, this one is E74, this one is W115. So each peak we know exactly which amino acid it is representing, and these this MWL, these are the amino acids, one letter code of the amino acids. So and then we get the structure, but it's very tedious task. It requires several months of work to get the final structure of the protein. And usually NMR structure are in symbol of 15 to 20 chains. So here, and using this NMR, again, we have identified which part of the protein is being binding with any molecule. So, and again, we use simple experiments from HSQC. In HSQC spectrum, since we have assignment of each residue for each peak or each contour that representing which amino acid, and if you add some small molecule into it, and if these contours has some shift, then you know, okay, this is the amino acid where your small molecule is going to bind. And that's how we identify and do the functional studies. So using this HSQC, we have identified this region is a magnesium binding region of the protein, and this one is the ATP and AMP binding region. We have done experiments with lots of different methods, lots of different nucleotides, but the other has no impact on this. Only these two nucleotides are binding, and that shows the real thing, that how NMR is helping us to identify the function of the protein. So these are the simple structural characteristic. I would not go to it. And here another protein, which is galactitol specific components, is important part for the sugar transport system. And if this proteins has been inhibited or misfunction, then the sugar cannot be transported in the cells of bacteria, and then bacteria, the ultimate would be the death of the bacteria. So these are important. And again, these are the same number of experiments similar that we have recorded. And this is the representation of proton NMR. You can see it's very much different from the other, but just get all the information. And these are the HSQC, which shows that each amino acid has been assigned. And this is a ribbon presentation of the structure, but definitely structure takes the backbone and the noses information, and then we get the structure. I can spend whole day how to get these done because it's a long task. And these are the presentation in which you can see these are the side chains in each amino acids. Some are pointing inward and some are pointing outward. And these we can really see each hydrogen, each carbon of each amino acid side chain and backbone. And here, this is the different amino acid. If we compare with the Staphylococcus aureus resistance and non-resistance one. And important part of this characteristic is Ramachandran plot. It's the very old, but is still being used, and it's the most important one. When you see that all the residues lies like 78% in the allowed region. In the Ramachandran, they have like alpha, C alpha, and C beta angles, which is being calculated, and then it's these all using these dihedral angles. Its plot has been assigned that if the structure is being calculated correctly or not. If not, then you have to work back to your assignments. And it's alignment with the already available structures with the in PDB protein data bank. You can see these are even with the highest one is not very much aligned, which shows that even if there are similar kind of residues, the structure folding could be very different. And this is another representation of a protein, which is a phage-related protein. And this is the first structure. And here we have done different sets of experiments. We have recorded FC, which is 4D and 5D experiments in NMR. These are actually experiments. With these experiments, we can get the information in the form of the peak list. We have to generate the peak list. So, and then we use these information for the assignment of protein. And again, these are the important criteria. And it's, there are different representation, which shows if we subtract C alpha minus C beta chemical shift and plot it against sequence of the amino acid, then you can see those which are on the upside are the alpha helices. So there are 2 alpha helices, which we can see here in the structure, and there are beta sheets which is downward. So, and Here is another plot which shows there are noses, which are in alpha helices, since they are very, very jammed and close. So there are different NMR experiment which actually shows how these proteins looks like and how it works. So with the with more simpler one, we can again, this is HSQC, in which we can identify this protein was again the magnesium binding protein. So it's very important and most easy experiment in NMR, in which you have to have a sample of protein in your NMR tube, and you titrate, like the titration experiment, add some molecule into it and record HSQC, add some other different concentration and record HSQC. So it's like a titration experiments. And this is the more very simple but very informative experiment that can easily be done on any NMR spectrometers. Little bit like at least 600 would be required. So it's another example of the protein, which is from Klebsiella pneumoniae, and here it's a small protein with 83 amino acids, and here you can see these are the HSQC and proton NMR shows it's very well folded protein beautifully, and but when we get the structure, then we identify this fold is never been reported in the protein data bank. So you can think that the diversity, this 20 amino acids are being given to protein structures. So that is why it's still, getting the NMR structure or protein structure. It's not only very important, but it's a very tedious task, and it has lots of bottlenecks. If you are working on NMR and you got a good construct and you see, okay, protein expression is very high, very good, and then you can then when you check it on oligomeric state, and if the structure is oligomerized, or protein is oligomerized, then definitely you cannot go to the structure or using NMR. But X-ray, you have more flexibility, but they have a constraint to get the crystal of the structure of protein. So it's again a bottleneck. So we have lots of bottleneck that we have to pass to reach to the time, or that we can get that this protein can be leading towards the structure. And so these are the different representation. Here you can see these are the 310 helices, and there are like sandwich of beta sheet is there. So, and we can, this is again a representation in which you can see this part is little bit flexible part here. Even in here, you can see this each, because NMR is usually ensemble of 20 chains. And here you can see these are quite flexible, which is similar, you can see here. So, and the other is the RMSD value, the root mean square deviation is very good. So it's an important, and here now it's another representation of the protein surface, where you can see the electrostatic charge of the protein. There are positive and negative charges, which are representing here with the blue and red color. And here it is a representation of the side chain. And here you can see this side chain is so flexible in some part. Like here you can see this are usually the C terminal and N terminal are quite flexible part, and the other are quite much overlapped to each other of the 20 NMR conformers. And so this part is, I would say, it required lots of expertise, and This is something that I learned in Professor Bert Wittridge's lab in Scripps Research Institute. And with this, I established the replica of his lab in Pakistan at the ICCBS, where we do everything. We have everything that is there in the lab, in his lab at Scripps. So another part, because NMR is my expertise, so another part of my presentation is the repurposing of drug. You know, it's an important thing that for us, it's not easy if as a developing country, if we think that we can identify a drug and we can perhaps spend cannot be done in our lifetime. We don't have that much money available. And so repurposing an important approach. Our molecular bank, we have more than 600 drugs, and drugs mostly like FDA approved or other drugs that are really in the market. And repurposing is something that the drug is being used for treat something, but we can identify its purpose for some other treatment. So this is called repurposing of the drug. So we have been working on SARS-CoV-2 main protease since the emergence of COVID-19. And yeah, so we wanted to repurpose some drug using NMR spectroscopy. And as we understand that NMR is something which requires time. The experiments cannot be done except the proton NMR. Otherwise, the experiments can be time-taking, depends on the amount you have and the sensitivity of the nuclei. So, but what with the Professor Iqbal has suggested us that we have to use NMR for as a kind of, not the high throughput, but the medium throughput screening. So with the guidance of Professor Iqbal, what we have done, we recorded the proton NMR of all the drugs that we have, and we try to group into, he has actually given us task to make a group of 10 drugs. So far we have achieved 5, but we are trying to group it into 10. You know, the grouping in a way that when we record spectrum, the signals should not be so much overlapped. So we can identify the signals of each molecule. And he asked me to make a group, use fast techniques to identify that which of these drugs is binding with the protein or any protein that is related to any disease. So we have targeted SARS-CoV-2 main protease. And as we know that with the infections of SARS-CoV-2, when the viral RNA translate, so there are polypeptides that produce. These polypeptides is being, with the help of the AMPRO, SARS, which is also called main protease, and the PLPRO. There are two proteases in SARS-CoV-2, which actually proteolyze these peptides and make it different small fragments, and that is being used by the virus for different purpose. So if we inhibit AMPRO or PLPRO at this position, means we are stopping the growth of the virus in the cells, because infection has already been done here. But if we inhibit AMPRO, the main proteases, or PLPRO, So these two proteases can stop the growth of virus. So this is the main target. And what we have done, we use the drug repurposing approach. So, and we use in NMR, there is a fascinating technique, which is saturation transfer difference NMR. You know, it's very simple and it can be done easily on 400, 500 megahertz also. It doesn't require very high field NMR. With protein structure, we have to go to the high field because otherwise we cannot get the nosy, good nosy, highly resolved nosy that can use for the structure determination. But this is the technique in which the protein requirement or any enzyme requirement is very less. It's just we can use like 1 or 2 micromolar of the protein, and we can use it to identify a small molecule that can bind with the protein. So what I will explain here, the saturation transfer difference NMR, in which what we do, we record NMR spectrum. There are 2 spectrum recorded. One is off resonance and other is on resonance. On resonance means we are saturating protein signal. So usually we select, we have to record a proton NMR of protein, which is not labeled protein, which is available, at the minimum cost. We saturate protein and identify which the good part of the protein, which is used, which can be used for the transfer of signal. So what is actually happening here? So when we have, like, for example, a protein, and if you have ligand, so usually the reactions are forward and reverse reaction. Okay, a molecule go inside and come out and the other go inside. So this is how usually two-way reaction takes place with any protein or enzyme with ligand, unless the ligand bind and just kill its property and cannot come out. So otherwise it's always like a molecule going in, coming out. So what we do, we record, if this is the protein, we identify a proton in the protein, just a proton, we saturate. We give a pulse to that protein, a proton of the protein. Since it's very big molecule, so spin diffusion takes place very fastly. Because, any protein, a proton cannot take and keep the energy level to itself, it just need to give to the other. So spin diffusion takes place and immediately goes to the all part of the protein, and definitely transfer to the part which is binded, transfer to the part of the ligand which is binded with the protein. And we then subtract this spectrum. That's why it's called difference NMR. So we have 1D NMR and then of ligand. And when we record on resonance, which is with the saturation, and we subtract these two, like on resonance and off resonance spectra, and we get a difference spectra. A difference spectra shows only those part of the ligand which binds with protein. So it doesn't give the information which part of the protein is involved in binding, because we are not studying protein, we are studying small molecule. This experiment is doable here as well. So you guys can easily learn and do these experiments here using NMR spectroscopy, because it doesn't require very high fields. It require, because it's a ligand-based experiment. We are studying the part of the ligand which binds with the protein. So here, with the guidance of Professor Iqbal, we have make mixture of 31 mixtures. So this work is being continued. So I just want to explain that so far we have evaluated compounds like 150 compounds in 31 mixtures, and this is amazing guidance of our mentor, because if I think of running 155 STD NMR experiments with one experiment taking 1 day, so think about how much time is required. But we record only 31 NMR experiments, each experiment one hour. So this is like medium throughput that Professor Iqbal has guided us that we should use NMR as a medium throughput technique, because NMR requires time because of the sensitivity issue. And whatever using these mixture analysis, when we identify in the mixture that there are some protons of some molecule or drug that are interacting, and we can see in the difference NMR that only those shows the difference which are interacting. Then we record a full STD experiment of 12 hours or 13 hours. So a good quality spectrum to report. Otherwise we got the information, okay, these are interacting, and we can, but for the sake of good quality picture and good signal to noise ratio, we would like to record the full of a single. Then we record only for those molecules, STD, full spectrum, which only those which are interacting. And in this way, you can think about, we have identified only 10 compounds out of 155. So instead of running 155 days experiment, we spend time of 5 or 10 days. So to run these experiments. So it's like medium throughput, and we have used this technique. And then we identified those which shows interaction. Then since Dr. Iqbal also shown that we have a lab now, which is BSL-3, in which we can propagate virus. So one of my colleagues, Dr. Sabah Farooq, is propagating SARS-CoV-2 in viral cell line. So she has used only those molecules that shows an interaction to identify if these molecules or drugs are really have an interaction and killing the virus itself or not. So you can think about that, starting from nothing, we could identify there are 10 drugs that are interacting, and these drugs we got inhibition studies of this virus as well. So we use NMR as a basic tool for drug repurposing. Okay, so these are the molecules that we identify. I would like to show, and this is the protocol that my colleague has been using for the SARS-CoV-2 propagation and the inhibition studies. And these are the images, because these viruses has been grown in the viral cell. So this is the control cell, and this is like infection of the cell with virus after 24 hours, 48 hours, and 72 hours. So you can see these are plaques growing. So that's why it is called plaque reduction assay. That assay, and then the experiments is being used like real-time PCR to identify it's really has an effect on the virus, identifying using different type of gene target. So this is these are the drug which has already been reported, and people are using for chloroquine phosphate for the SARS-CoV-2 infection. We have used to see the validity of our experiment, and we could see this part of the ligand which is in color, and we can see here this is the one which is binding with the protein. So using STD NMR experiment, because this is the proton NMR, and then the next one is the difference NMR, the STD difference, the situation transfer difference NMR. And then we have done the plaque reduction assay, and we could find that at 41.5 micromolar concentration, this is 63.2% reduction in the plaques of viruses. And similarly, remdesivir, which is also being used as an antiviral drug for SARS-CoV-2 infection. And we have done this STD of this one as well. And we could find out, those which are in color, it shows different level of STD effect. And we could see that it shows 86.7% at 82.3 micromolar concentration of this compound. And similarly, dexamethasone is a steroid, and we could find out that there are some protons in the S difference NMR. So you can see these are the upper one is just the proton NMR of the ligand. And here is a difference NMR. We cannot see the signal of the protein here. First, the concentration is very low, and we use in the pulse programming different pulses to minimize the signals of protein and protein protons. So that's why we don't observe signals here, and we can see there are the part of the ligands of which is binding with the of with plaque reduction 2% inhibition. And this is a drug which is dietesium hydrochloride, which is a calcium channel blocker drug and used to treat hypertension, high blood pressure, chest pain. And when we have done the STD NMR experiment, we could see that methyls and the third astromethyl are the one which is binded with the protein, which is SARS-CoV-2 protein. And with the plaque reduction assay, it shows 81.8% reduction with 82.3 micromolar concentration. And we have done this docking. As I told you that as a chemist, I don't that much have a belief on docking, because I have observed that whatever you have, you want to dock, you can dock, even you don't see it in the real spectrum or in real assays. So, but it gives a beautiful picture. So for the sake of beautiful pictures in our publication, we do this docking. But we do it because we know there is a real interaction, because we have observed using NMR spectroscopy. We have the result of the bioassay that shows this has an effect. So, but we use it, to have good pictures in our papers as well. That's what I feel. And so it shows that this protein, this molecule has an interaction with different amino acids. You can see it's a beautiful, nicely image, and this has some binding. So it's very simple docking methods that we use. And another thing which we have used in computational in silico studies, this is simulation studies, which actually shows that this protein ligand complex is very stable. So it just not, if this IMSD value is very flexible, then you don't see a good simulation. If the simulation is good, so it means the protein and ligand. It's also in silico, but it shows, depends on the time you give, how much nanosecond you give for this computational study, which shows that this is a stable complex. And another drug, which is the mefenamic acid, which is a non-steroidal anti-inflammatory drug and used as a common painkiller. And we identified this is also interacting with SARS-CoV-2 main protease. And these are the red and blue color. These are the two protons that has an interaction, two methyls has an interaction that we can see. Only the difference, now you can observe in the difference NMR, there are only two which is appearing here, which has an interaction. Those protons which has no interaction gone. So this is why the difference NMR. We record the proton NMR and then we subtract from the, on resonant, off resonance spectrum, and then we got the difference NMR. And it's a real interaction which actually takes place. And then we have done the plaque reduction assay, and which shows 81.8% reduction at the concentration of 81.3 micromolar. And this is again beautiful image of docking, which shows it has interaction with protein. And this is simulation studies, which shows this ligand and protein complex is quite stable. And another drug, which is losartan potassium, and this being used as a angiotensin receptor blocker and used to treat high blood pressure. It also gives a good interaction. You can see those parts which are interacting are colored here, and like H8, H9, H10, H7, and these are the parts which are interacting. And this is this shows like 52% reduction in plaque reduction assay. And the docking again shows the nice representation. That's how this molecule is interacting with different amino acids of protein. And this is the simulation plot. And another molecule, which is amantadine hydrochloride, which is used to treat the symptoms of Parkinson's disease as well. So here, this is a small molecule and it's interacting, almost the whole molecule interacting with the protein. It shows 77.2% inhibition at 82.3 micromolar concentration. And this is the beautiful molecular docking image. And this is the RMSD plot, which shows that protein ligand is quite nicely has interaction and stay longer with each other. And here it's we have done using real-time PCR. We can there are amino acids which are always those amino acids which are hydrophobic in nature. They tend to go inside the core of the protein. They don't want to have outside to the surface because they are hydrophobic. They don't like to be with water or some buffer. So they are usually inside the core of the protein. So if we heat the protein and we have some kind of dye, a fluorescent dye, which can interact with those amino acids which are inside the core. So when we heat, the protein unfold and those amino acids are exposed, and the dye has an axis to bind with it, and we observe the change in the color of dye. So in this way, we can identify the TM, the point in which the protein start melting, and then we can, using this point, we can identify if the presence of the drug or molecule which are interacting, if it reduces the TM of the protein, that means that it has an interaction and it's helping to for protein to be deformed. But this is not the only way of a molecule can interact. There are many ways, but this is one of the ways that can be studied using TM using real-time PCR. So we have identified this is the red color is only the protein, and these are the different drugs which we have used to check the TM of and how much they are changing. So another part of my one of my student has started working that on the NS5, non-structural protein 5 of dengue virus, because dengue is our teacher, Professor Iqbal, again, he is the main source for us to guide in the right path. He asked us have to work on the disease that are our problem. So as a developing world, because the developed world never pays attention to those diseases because they don't get money for it. So we have to use our resources to find out the solution of our problem by ourselves. So dengue is again a big problem. We always, every year we have the spread of dengue virus, and many people died because of the dengue. So what we have used again, the approach of drug repurposing, and this is, you know, a statistical data, but in like 2021, there are 48,906 cases in dengue reported, and there are many who are not reported because the developing country people sometimes doesn't go to the doctor even, and 183 deaths. So NS5 is a non-structural protein, and it's main protein, and we consider if we can inhibit the function of this protein, then we can control the replication of virus. So it's an important target for the drug discovery. And here the NS5 consists of two domains. One is RDRP domain, and the other is MTase domain. So these are the two domains, which is RNA-dependent RNA polymerase and MTase. So these are the one, but we have the whole construct of, it's a quite big protein of more than 100 amino acids. And we use, again, simple STD NMR method to identify ligand of this. And this is the way it is shown here. And I would like here to explain there are limitations of STD as well. You know, if because it depends on, it's a nice representation. If you saturate the protein here, and once you see that there are molecules going inside and coming out, and if you saturate the protein, it shows the red color. And the molecule, it's like one molecule is going, getting the saturation, and the other is going. So you need to have like quite balance. If a molecule goes and stays inside, we will never observe an STD. It doesn't mean that molecule is not interacting. It means that we cannot identify it. And if their exchange rate is super fast, more than the NMR time scale, again, we cannot. So there is limitation of STD NMR as well. So here you can see this molecule is going, and we can subtract and the spectrum, and we can see only those parts, as we have seen, those part of the ligand which are interacting, and we can identify those. So here we have evaluated 75 FDA approved drug in 15 mixture which are interacting with NS5. And here we can identify diclofenac sodium, and which is a drug, and identify these molecules, these parts, which is the upper part is the difference NMR spectrum, STD difference NMR spectrum, that these are the protons which are in different color are binded with the protein. And again, this we have done some docking studies to shows an interaction of this molecule or drug with protein. And here another STD spectrum of etopride, and the upper one is the difference NMR spectrum, and we can see these are the protons which are in different color, and they receive different level of saturation. For example, we have the orange one shows the 100% saturation. We consider it's 100%, and then actually with keeping this at 100%, we have find out, okay, these are the other one have different different digits of presentation, different level of presentation, percentage of saturation that they get. So we have to choose one based on the highest one, that as 100, and then we use other as it's not working. Can I go here? So, yeah, so here we have used a docking again, which we can see that this molecule is being working with the interacting with other. I use this one. Okay, so and here another molecule which is etinolol, and which is used to treat high blood pressure. And here also you can see this in the different NMR spectrum. There are different level of saturation that we got. Sometimes this is almost the whole molecule is binding with the protein. It's a small molecule, it can go to the active site and you can see the level of binding almost all the proton, but definitely there are some which are more closer, receive more saturation. So we consider those as 100% and then we optimize the other accordingly. And the molecular docking studies shows that this ligand is binding with the docking site of the protein, with the binding site of the protein. And another molecule which is scopolamine, and here we can see this is the proton NMR. So proton NMR is always there because we want to show the difference. What is the difference between the proton NMR and other molecules? So this whole molecule is also interacting, and this is the in silico studies. And here we can see that these are the the protons which shows the interaction. Here you can see in orange color proton, this one shows the 100%, the green color here, these two show the 62%, and this one, the blue one, shows the 49% of saturation received from the protein. So we are not saturating the molecule itself, we are saturating protein, and the saturation is then transferred to the small molecule through interaction, because it shows this molecule is interacting with the protein. And then we have checked and we find that polymerase inhibition activity of with the diclofenic acid, we got this result is still we are doing these experiments. So diclofenic sodium we identify using STD NMR, which shows good protease inhibitory activity, and at the concentration of 500 micromolar and above. So this shows its result. And I would like to show a brief here that using NMR spectroscopy, we can identify the function, also the reaction monitoring, as we have read about it. So we have a protein, we have expressed this protein and or synthesized this protein, and in our lab, which is NADPH hydrate dehydratase enzyme. And this is an important enzyme for the DNA mismatch and metabolic proofreading. And here, this is the main reaction which is performed, like NADH is converted into NADHX with the expenditure of ATP. ADP is converted into ADP, and here this bond, here you can see this, the hydration, the blue color shows the hydration takes place. The OH group is being added, and this can be here with the, and this is S and R confirmation of this molecule, and this molecule can be either one of them, can go to cyclic NADHX. So this NADH conversion to RS or cyclic NADHX in the acid environment is being catalyzed, specifically the dehydration of S form is catalyzed by NADPH hydratase dehydratase. So this is an important part so that we can identify the reaction. So we have used, we initially we were trying to do it using in the lab, but then I asked my student, this one is being done by a student Uzma, and then I asked her that we have to use NMR tube for this reaction, why we are doing on the lab and then transfer it, and when we have to just read NMR. So we will do this at the NMR tube. So, and we can increase the temperature of NMR up to the boiling point of the solvent we want to use. So there is, NMR is very flexible. It's a big protein. So here you can see, this is the proton NMR of in which S, R, and cyclic shows the difference. So S has this kind of, and the R is, R has this, and S has this kind of a spectrum, and cyclic has this. So it shows that if we have this molecule, we add into the NMR spec, and first the protein, and then we have heated up in the acidic condition, and in the NMR tube, we have done only in the NMR tube, then we could identify, okay, that the reaction is functioning, that this molecule in the presence of an enzyme is being cyclized using the NMR spectroscopy. So with the difference spectrum, we can easily observe. So this I wanted to show, and then we have done some STD NMR experiments as well. And to see, here I would, at the end, I would like to explain that the only thing is NMR is a robust technique. It's we can use it as a medium throughput, and especially the second part, the first part which I showed, it required high field NMR spectroscopy. You need to have at least 800 megahertz because you need a good NOESYS experiment. The backbone can be recorded on 600 with a cryoprobe, but NOESYS you need a high resolution. So, but for this, the other part, it's can be done easily with the small spectrometer, like 400 and 500 spectrometers are also fine, because we are not studying signals of protein, we are studying signals of a small molecule, and that can be easily studied using small magnet. So this is one of the great strengths, and I would say, you know, I learned NMR spectroscopy from my mentor, Professor Iqbal. I've been taking his lecture at least 10 years. So it's not an easy technique, and he is an amazing teacher. I would say that he is the best NMR teacher. He gives the concept, the hardcore concept of not the application, but the backbone, how the signals are generated, what is the theory of NMR. So he is the best teacher, and he just gives the concept and things that just goes in your heart and mind, and you never forget. So, and now I would like to acknowledge my students, Dureshawar, Faisal, and whose protein have been working. They have synthesized the protein, and for drug repurposing, Abdul Mateen, my student, has been working on this, and for AMPRO and for dengue viral NS5, Dr. Asmatullah has been working, and we got this construct, Professor Paingong, and we have been financially supported from different projects. And I would like to also show you here a brief pictures of our institute, NMR, because I'm in charge of the NMR facility. This responsibility has been given to me by Professor Iqbal. And we have 13 superconducting NMR spectrometers, and I would like to show you here. And we recently have upgraded all console to the new console, NEO, and this last in 2021. And now the electronics is amazing. The sensitivity is really high for all our spectrometers, and we have 3 cryoprobes with 800 with the cryoprobe is a sensitivity about 1 gigahertz NMR spectrometer. The resolution remains same, but the sensitivity is very high. And we have 2 600 with the cryoprobe and 1 500 with the cryoprobe. So 4 cryoprobes And with the 600 cryoprobe with the 600 spectrometer with the cryoprobe, the sensitivity is up to 750 megahertz. So these spectrometers are really amazing, and whoever is in our institute, all students has to run their own experiments. We have some, and this being initiated by Professor Iqbal. He asked that our technicians should not record the spectrum for students. So we are serving as the hub for our national service as well. We got samples from all over the universities, different universities of Pakistan, so that our technicians run. But our students' samples, they have to run by themselves, because this is the only way for them to run, to learn at this level. And we have different type of probes as well, inverse probe and multi-nuclear probe. We can detect all kind of nuclei using multi-nuclear probe. And we have various different type of probe that you can see here. And another thing that I would like to invite all of you, that with comstech, ICCBS, we have NMR spectroscopy user group. So there's a form here, and I would share to Professor Solomon as well, so he can send on all of you. And it's free, and you receive the NMR information and related to NMR spectroscopy, and some lectures and some conferences accordingly in our newsletter. So we have comstech ICCBS newsletter as well, monthly. So we send this. So I would like you to join our user group of NMR, and then we'll be in touch with you. And here there are different services that is being offered by the ICCBS. These are, all NMR experiments, not only this, whatever in the library of a spectrometer, Bruker spectrometer, we can do it. And we have been recording these, so we can help our brothers here also. And these are mass spectrometric and other facility that being offered by the ICCBS. These are the bioassays. All these bioassays, we are doing this for our as a national service to other institute with the initiative of Professor Iqbal Chaudhry. And thank you so much for your attention.
The chair of the session is there. Professor Kerub, are you there? Hello?
Anyway, we can take some questions, okay? Yes. You can come. And any question from the online attendees?
Thank you so much for the nice presentation. I have a couple of questions. When you're working with the SARS-CoV-2, what level of safety were you working on purifying that proteins? Second question was on dengue. We have 7 non-structural proteins. Why choose NS5? And in this technique of doing the binding studies, do we know the active point? Have you solved the structure? I know there's X-ray crystal structure on this, but do we know where it's binding these molecules? And the last question, this is on the cell. You're talking, these things are being released on the cell level. So these small organics, how do they reach the cell so that they can?
Thank you so much. So I like, so one by one I will answer. So the first question, the level of biosafety when we are working with the SARS-CoV-2 AMPRO. So this protease, when we have the DNA, it's absolutely fine. It doesn't require high safety level. It's a normal lab that we can work. So once it doesn't require means like BSL level 3 or something, but for the propagation of SARS-CoV-2, the virus itself, we have BSL 3 labs. So as everything is being working in BSL 3, but once we have DNA, because it's a construct, it's a plasmid, it can be used in normal labs. So it doesn't require any means normal lab safety, not like BSL 3 level safety it requires. So, but for propagation of virus, we use BSL 3 facility. And second is your question about NS5. We have choose this one because it's an important target for so far we have studied, and we have a plan to work on others as well. So it's just a starting point that I recently assigned one of my students to work on this non-structural protein 5, because it's has two important parts like AMPase domain and RNA-dependent RNA polymerase. So I feel like this is the very important part. So if we can inhibit this one, so it can inhibit the life cycle of the virus. And your third question was how if we can identify the part of the protein or in which the ligand is binding by using proton NMR or STD NMR, we cannot identify. This is what I was explaining, because it doesn't require very high field NMR, because we are studying which part of the ligand is binding. We are not studying where is going to bind. Okay, so it doesn't show if it is inhibiting or not. So that is why we identify we do the bioassay afterwards. First, we find out some interaction. Instead of just doing here and there, we use NMR spectroscopy as a tool for medium throughput screening, and then identify which part of the ligand, or if the ligand is binding or not. You can see out of like 75 in case of this, we have only a few drug molecule which shows an interaction. Okay, so in this way, but we cannot identify which part of protein. It requires high field NMR spectroscopy. And again, NMR has a restriction. We cannot go up to like 35 kDa protein. It's a big protein, more than 100 kDa. And again, if we use X-ray, so it has its own limitations as well. But we did not use my field as NMR, so I have to focus all the technique using NMR spectroscopy. And your last question, how it will reach to the cell, and so we have usually these molecules, because these are drugs. We are trying to repurpose the drug. So the drug, this is the beauty of this one, that all the safety and efficacy and everything was already been studied about the drug. And if we can identify some other function for which it's been reported, then this is the only way we can contribute in some drug development process.
Thank you very much. So I can ask, and first of all, thank you very much for introducing us to this fascinating world of protein NMR. When we see an NMR of a big molecule, we get really scared. I can imagine how complicated the NMR spectra of a protein, a large protein, will look like. If you look at the PDB, the majority of the compounds are generated from X-rays, using X-ray. Given the right size, as NMR has information on the size, given that which method we prefer, or which method would you think be better? The NMR generating structure, or let me put it the other way. Because NMR, despite its limitation, I think it is universal, because you don't need a crystal, isn't it? You don't need the structure. So given choice, which one would you go for to determine the structure of a protein?
Thank you so much. Nice question, but you answer by yourself also. Because NMR is the technique in which you can study near the physiological conditions of the protein. So this is an advantage over the crystal, because in crystal, you cannot go up to that level that the NMR you can go. And definitely NMR has more limitation as compared to X-ray. X-ray has limitation of making it crystallize. And for this, there are robotics also available to check like millions of conditions at the same time. But NMR has, this is the fascinating thing in NMR, that you can study at the molecular level, and you can study the proteins which are not folded, which are flexible. You can study in NMR, but those flexible part in the X-ray, they are disappeared. Yeah. You cannot get it. And definitely it's very complicated method. That's why I require 3D NMR at least. We cannot work only on 2D. 2D is just the basic of all this structure. And it required quite time to get the right protein that can lead to the structure. So there are lots of bottlenecks in NMR because not only the size, the protein has to be soluble at require high expression. We need at least 5 or 8 milligram per ml protein if we want to get the structure, which is very high concentration, and it need to be soluble at this concentration as well. We don't want to have aggregation. It need to be stable for several weeks so that we can record the spectrum. It requires recording of several weeks time because it's 3D spectrum. And there are some experiments that are very insensitive, that you to see these carbons and protons, you need to do lots of different type of experiments. So it's fascinating, but it has its own limitation. X-ray has its own limitation, but definitely X-ray has less limitation as compared to NMR.
So I can imagine you have several thousands of peaks. Mm-hmm. So do you use any kind of algorithm to annotate, for example, the peaks for the different amino acid residues? Do you use any database?
No. It's actually no. That's again the thing that there is in BMRB, which is biomolecular NMR spectroscopy database, in which everyone who is recording NMR, they have to deposit the structure. It's mandatory. Without this, PDB doesn't accept the structure. So they just make it mandatory so that they can increase but it's very different because every protein, there is a range, like C alpha, the first carbon after the NH, or it's which the range is from it's 52 to 60 ppm. But it can lie here and there. It's fine. It's a little bit changed in the chemical shift. So there is no such kind of database that we use to assign this. This require, depends on the interaction and how the protein is folded, because it's a microelectronic environment. So this microelectronic environment changes with the folding of the protein. So it's not like X-ray that you can get this similar kind of a structure and then you try to fix your structure on this. You have to assign each and everything if you want to do it. Even it's reported and you want to do it, you have to assign. Yeah. Thank you. Yeah, I didn't find the question.
Yeah, it's not a question, but
yeah.
Yes.
Yes.
You are repurposed some developed drug in only no one is in where the mechanism of action was not known. And discovered acting as sample. Would it be possible there to discount, to use that model, to discount the effect of some some multifactorial effect of an extra using that model? Using NMR?
I did not get your question actually, I'm sorry.
There is this legal model somewhere. Among the drugs that
Yes, yes. Yeah.
You didn't know which protein is there.
Yes.
Take it from there. You might have a multifactorial extract with several compounds.
Okay, I got it now.
Can you use that model?
Yeah.
To identify this?
Definitely. Yes, definitely. Okay, so we have used, as I show you, we have identified the mixture. We have made the mixture intentionally by ourselves, a mixture of 5 drugs, and then check it. So you are asking about if instead of this mixture that we made, we use the mixture made by plant. So, and can we use it? We can definitely use it. And we can identify the peaks which are interacting. But even then, you have to identify these molecules. If you identify because if you take a mixture, then you don't know the composition right now. If you know the composition of the mixture, if you know, then definitely no problem.
All right. I have an extract. Very simply prepared.
Yeah.
With at least 30 compounds.
Yes.
And several intermediates.
Yes.
And division activity.
Yeah.
Over 95%. That model tell me for among the certain known compounds there. How do you check?
Yes, definitely. We can definitely do it. And if more than that, if you know the composition, if you have the compound, if you have like a standard compound, like you know that this was the one, and you identify in the mixer, you can add those compounds intentionally, to see if it is really interactive, because that one has a more concentration as compared to before. Or you can separately check it and overlap the spectrum of one over the other, and then identify, okay, this is the real one, this is the molecule which is interacting, and this is from the extract. You can definitely use it. Yeah.
Thank you. I don't know if Professor Kiru, are you able to unmute?
Yes, I'm able to. Yes.
Okay. So you can now officially thank her.
Okay, yes. Thank you very much, Professor Wahab, for an excellent and informative presentation. I'm sure many students from chemistry have seen that you can actually be an organic chemist and also do other diversify, maybe to structural biology like you've done. And they should not be shy to do so. So I've also seen that we have very good, the labs you have are state-of-the-art labs. And the research you're doing is also state-of-the-art. So I'm happy with the collaboration MOU that we have formed. And I hope we are going to send students there to do the same research. So thank you very much again. And we hope to continue having a conversation. So this is not the end.
Thank you so much. And I'm again very thankful to NAPRECA and comstech for giving me this opportunity to present my research at this forum. Thank you. And here I just want to answer a short question that I just seen, that which software have been used for docking and simulation. So we have used Schrodinger. Thank you.
Thank you very much.
Thank you very much. So I think then we are going to have Professor Said Ghulam Musharraf coming to present his work on metabolomic tools for profiling and fingerprinting of phytomedicine. But before that, let me just give you a short introduction of who he is. Professor Said is from HEJ, Research Institute of Chemistry, ICCBS, University of Karachi, Pakistan, also, just like Professor Wahab. He is a well-known scientist of Pakistan in the field of bioanalytical mass spectroscopy, spectrometry. Currently is the author of over 200 research publications in reputable peer-reviewed journals, and he also has one US patent. He has supervised over 26 PhDs and 40 MPhil students in the field of mass spectrometry. And due to his scientific contribution, he has been awarded with several national and international awards and honors, including Civil Award of Pakistan, Gold Medal Award in Chemical and Pharmaceutical Sciences, Dr. Ataraman Gold Medal Award, just to name a few. He has also received several cash awards in various categories. His presentation, as I've already said, is entitled Metabolomic Tools for Profiling and Fingerprinting of Phytomedicine. Welcome, Professor Said.
Thank you very much. Am I audible?
Can you speak a bit louder?
Yeah, am I audible now? Yes. Am I audible? Can you hear me?
Yes, reasonable. If you just speak a bit louder, and then we'll.
Okay,
the ones online actually can hear you very well. The ones in the hall, just a bit, but it is okay. It is okay. It's okay, Prof. You can go ahead.
Is that okay?
Yes, yes.
Okay, great. Once again, thank you very much, Chair, for your kind words. I'm very much thankful to the NAPRECA and the comstech, particularly to our beloved teacher, Professor Dr. Iqbal Chaudhry, Director ICCBS and Coordinator General comstech, for giving me a chance to share my research findings at this forum. So I think the formal lecture was more about the NMR. So I'm just going to talk about the sister technique, which is the mass spectrometry. So in more detail related to its application in natural product chemistry. So I think since last 2 days, you are listening the importance of plant, that the plants, different plant species are known for the generation of unique and novel drugs. And since last several centuries.
Professor Said, have you, sorry, Professor Said, have you opened your slides? We are not.
Yes, I've already opened that and shared. So do I need to?
We are not seeing, the only thing we see is the folder that has your presentation.
Okay.
Yeah,
Wait.
yeah.
Just a minute, please.
Again, another very fascinating field, protein chemistry, metabolomics. We are learning a lot.
Yeah, sometimes, okay. There is some problem that is not getting shared. Okay. Share screen. Can someone guide me that how I can share it, because I was trying but it's not able to open my file. And should I go to the share screen?
Yes, so yeah, you go to the share screen, and it
Okay.
should, when you do, the screen should be populated, and it should allow you to see where your slides are. But in case it refuses, let us see.
Yes.
In case it refuses, you can also email me just as an option.
Actually, when I'm going to share the screen, that or the possibility, there is no option of my files. I mean, there are some different options.
Close the sharing and redo it again. It will probably populate this time.
Screen. Okay, wait. Maybe I need to open this first. Yeah, probably this is how I can do that. Right.
You can also send me an email with your slides. I can also try from here in case it just decide to.
Is that okay? Is that okay
No.
now?
Yes, yes, finally. Yes, thank you so much.
Okay, great. So yeah, sometimes you need to learn all these things. Okay, so I was talking about the my mentioned the importance of plant. The plants have been the source of different biomolecules since last several centuries. And there are several drugs and drug lead molecules have been identified from the plant species. And the people are using them for the treatment of different disease. So this is the motivational source that why the people are still working on plants for the identification of new molecules. But that the problem associated with especially plant, for those scientists who are working on the phytochemical analysis, there are several issues, but I just want to highlight one which is majorly encountered by most of us, is the redundant isolation of compounds. For example, in this case, you can see that the two most common compounds, the beta-cetosterol and gallic acid, these are the one of the few secondary metabolites that are present in the plant. And you can see that since last 20 years, around 4,000, over 4,000 publications are actually highlighted the isolation and characterization of the same compounds again and again. So it's a sheer wastage of time and money. So that is the major issue, that the redundant isolation of compound in case of different plant extract. So this is the major issue, that the redundant identification of compound. Another issue obviously related to the amount of the plant material that is being consumed. So because if you want to have some variable amount of a natural product, you need to use tons of plant material, which is nowadays quite difficult because of the different the sustainability issues of various plant species. Another important thing, if you are going to talk about the plant-based products, the herbal products, the phytomedicines that we have in our local market. So these medicines have several issues, and that's why they are popular, but not that popular where it should be. The first one is obviously the lacking of knowledge, the gap, because they are documented but not well documented where it supposed to be. Obviously, these herbal medicines based on the supply of raw materials, so there is always a problem of inconsistency. That is something always dependent on the field, on the production of the raw material, or maybe some other third party. So that is another issue. The third part, which actually quite difficult and is still not able to address in most of the herbal products of our phytomedicines, there's the standardization. The lack of, particularly in some case, the biomarker is not available, or sometimes chemical markers not available. So you cannot standardize the species in terms of their efficacy of the product. Uh, uh, Obviously, if you're talking about the clinical evidences, because they are being used since long, a particular plant species. When you're going to talk about a formulation, so there is a very few clinical trials of herbal medicines reported worldwide. So there is another gap that needs to be put in. The fifth most important part, particularly for the analytical scientists, that it's not that easy to standardize or to analyze the polyherbal formulations, because the polyherbal formulations consist of, in most of the cases, not only the one plant material but sometimes 5 to 10. So these are the major issues that are associated with phytomedicine particularly. So how you can address these, maybe some part of these issues. So what I'm going to talk about the use of metabolomics. So the metabolomics is a kind of field of omics field, which is since last 2 centuries widely being used to address the different biological and chemical questions. The metabolomics is not only with the plant-based, but for the biological system as well. And the major part of this application is actually being explored by using the different biological fluid. But as we know that the plant is almost have the same cascade, starting from the genome, then the transcriptomes, and then proteome, and the phenotype, that is metabolites. So at the last step is actually nothing but metabolites. Metabolites is actually represent the phenotypic state of an individual, of a healthy person, of a disease, or of a particular plant species. And these phenotype, these small metabolites or secondary compounds, they can be used to identify a fingerprinting of particular plant species as well. So this is the metabolomics where the people are using nowadays, in particularly in case of fingerprinting development and for the herbal standardization of herbal products. So that's what I'm going to talk about in 20 minutes. The tool that I'm going to talk more about, the mass spectrometry, because mass spectrometry is a technique of choice that is available and highly associated with the metabolomics analysis and the fingerprinting of plant material. And if you asked why, so the answer is here. You can see that the mass spectrometry is a technique which is truly available in a form of hyphenation with the detector, with the mass analyzers. Like you can see that the LC, the GC, and the CE, and these are the different chromatographic techniques, and smartly coupled with the mass analyzers for separation, followed by the analysis. So that is one reason. Another important, that the mass instrument offers a kind of hybrid approach, which means that you can couple more than one mass analyzer, like QTOF, triple quadrupole. So these are the different combinations available in the market. And because of that combination, you can particularly focus on a very small concentration of metabolites and selectively identify them in biological system in a complex sample. The third important point, the availability of a broad range of ionization technique, starting from the EI to the Marley, which means that you can not only analyze the very small non-polar compound, but you can also analyze the larger polar molecule compounds like proteins and peptides and DNA. So, I mean, it's a broader coverage of almost all types of analytes can be analyzed. It depends on what techniques you are going to use. On top of that, the new advancement of mass spectrometry is still coming in, and they are using very much in the biological system. And some of the mass spectrometry tools is used for during the operation of, the breast tumor. So these are the few glimpses of, the mass spectrometer, that why the mass spectrometer is widely being used for metabolomics and even for proteomics as well. But this slides I personally like very much because it actually tells you the exact beauty of mass spectrometer is nothing but the sensitivity. The mass is the highest, is one of the technique which has high sensitivity. For example, in one of the papers published, you can see the authors claim the detection of 800 yoctomoles. So you can imagine that this is the lowest level of the detection. But apart from that, the routine practices actually started from femtomole to the picomole. So it's quite routine practice that people are using that. And because of that, they are, it's commonly being used for different application sites, including environmental as well. So if you're going to talk about the analytical techniques available for the metabolomics, particularly in chemical fingerprinting of plants, so you can see that there is an option of NMR as well. So I'm not going to talk about the NMR because it's Professor Rati has already explained NMR nicely in an elaborated version. So, but if you just compare the NMR with mass, the problem is about the sensitivity. The mass is actually a kind of technique which is highly sensitive. And on top of that, there is a quick separation of compounds. A real separation of compounds is straight away connected with the mass spec, and so you can quickly identify with the help of database. If you're going to talk about the mass spec, particularly in case of chemical fingerprinting, there are always again two options. The one is GCMS, it's a gas chromatography mass spectrometry, and another one is LCMS. The GCMS is actually a kind of technique which deals with the analysis of mainly non-polar compounds. Polar compounds can be analyzed, but after necessary derivatization, which is actually a drawback of GCMS. But especially in plant non-polar metabolites like essential oils and some fatty acids, so and terpenes, they can be very efficiently split out and then analyzed by the mass. Similarly, in case of LCMS, there is an option that you can separate out from the medium to polar compounds very efficiently using mainly the APCI source or the electrospray ionization source, followed by their detection with high sensitivity. So that was a brief introduction about the mass spectrometry and its application. So now I'm going to talk about the some high throughput mass spectrometric methods that we have developed for the chemical fingerprinting of various plant species by taking few examples, because I have been allocated for 30 minutes, so try to finish that within that time limit. Anyway, so this is one of the examples where you can see that the plant metabolites can be analyzed efficiently in a high throughput way by using a two-stage protocol. So this approach that we actually proposed and published few years before, and what we do in that part, the first part, you can see that is the generation of chemical fingerprinting. So we used to analyze a very analytical amount of plant material by using HPLC ESIMS. So with the help of this and high resolution analysis, somehow we able to identify the presence of metabolites that is present in the species. The second stage, if you just look, is the quantification of the marker compounds or biomarker compounds, if it is already known. So the second stage, you can quantify exactly the biomarker components in the plant extract or probably in the herbal formulation. So this high throughput approach is actually gives you a complete picture, not for the qualitative analysis, but for the quantitative analysis. And we are very frequently using this approach for the quick analysis of medicinal plants and for development of their fingerprinting. So if you just see at the right side where I have mentioned the identification of compounds, that is the major issue I would like to say, unlike NMR, because in NMR, the compounds accuracy identification is quite accurate. The mass is not like that. The identification of compounds based on the mass spectrometric features. And most of the cases, in plant case, you don't have the standards for their accurate identification. So how you would make sure that you are going to identify the correct compound, but not the artifacts, not the false positive. So in addition with the methodology, we were quite strict in the identification of metabolites structure. And we applied this level of identification, the first level of identification, obviously based on high resolution data that we usually recorded. The second step that we actually compared this data with the reported data, which is very, very important. In most of the cases, I've seen that the people are not comparing with the reported one, which is not good. I would strongly recommend whenever you are going to develop a chemical fingerprint, please develop your own database first based on the reported literature. The third part, if your instrument is smart enough or having a kind of facility where you can develop the tandem mass. So this is another tool through which you can accurately identify the structure based on their particular losses of fragments and reduction of process. The fourth part, that after completing the data, the tandem mass, there is an option. There are databases available by which you can compare your tandem mass spectral features. And if you are really lucky, so you may get the accurate identification. This is the fourth level of identification. The fifth level, obviously, if you already have the standard, so you can use the standard as well for the absolute identification of. So this is the strategy that we are using in our laboratory for the quick dereplication of plant extract and chemical fingerprinting development. As I have mentioned that the identification of compound is really important, particularly if you are going to use the electro-spray tandem mass spectrometry technique, which is mainly for the medium to non-polar compounds. So these are the databases available where you can look. The very extensive data is actually from the NIST side, where you have millions of data, but that data is actually mainly addressing the EIMS spectra, which means that if the compound is non-polar, you can very efficiently analyze and identify with the help of NIST database. But when you are moving to the polar technique like ESI and ESI MSMS, so in that case, unfortunately, the available database is limited. There are some databases who claim that they have a natural product, tandem mass spectral features, but again, it's very much restricted up to just a few hundreds or sometime maximum 1,000 compounds. So that is the bottleneck or drawback, and where we need to contribute that how we can accurately identify if a person is going to use this electrospray or LCESI MSMS technique. We are fortunate, and I'm personally very thankful to Professor Dr. Iqbal and Professor Dr. Ataur Rahman, our legendary scientists, because they are well thought and they have a vision, and because of their vision, we are now benefiting. So I'm happy to introduce this word of molecular bank, because in our center we have a molecular bank. And what is molecular bank? I'm happy to explain. Actually, this is the bank which has a repository of thousands of compounds in our center, because our center is well known in the field of natural product chemistry, and since last several centuries, the students are purifying the compound and structurally characterize those compounds. So we started this practice since last several years, that every student has to deposit their few milligrams of the compound isolated and characterized into the molecular bank. And now you can see that we have over 13 to 40,000 compounds in our bank, fully structurally characterized. Some of them are synthetic, some of them are natural product, a few of them from marine source, from the other natural products. So, but this is the library, a very unique library that we have only, I say, because the other people are claiming, but they really don't have such high numbers of compound that we have. So we have, because of that library, I worked with Professor Iqbal, and we submitted a project for the development of the tandem mass spectral library of those natural products that we have. So around 3 to 4 years before that, we have started to work on the development of mass spectral database library. And this is one of the papers that I think just published last year, where we have discussed the pooling strategy, that how you can develop the database of natural product if you have a purified compound. So what we proposed a pooling strategy, which means that you can pool the different natural products in a one sample in order to make it high throughput. So the strategy is actually addressing the pooling of standards from 50 to 100, which means that if you have a one injection, so it must have at least 50 number of standards, reference standards, and it could be up to 100. And this pooling is actually based on two important things. The one is log P value of the compound, and the third on the masses. We try to pool those masses, those reference standards who has same masses, the isobaric compounds. So we just separated out. So these two strategy, the log P value and the masses, these two really works efficiently, and we developed a database of several natural products, and it's just in publication process. Like in the first paper, you can see that they just mentioned the pooling strategy. So this is the second, the continuation of the pooling strategy. As I have mentioned that this one of the paper we have published related to the analysis of alkaloidal pool samples. And what we have done, we developed a database of alkaloidal plant metabolome. And again, you can see this is the example of one of the pooling samples, and where you can see that up to 74 reference standards were pooled together, and you can see they were nicely separated out. And at the bottom side, you can see that their mass value, their calculated values, their ppm error. The ppm error is really good. And at top of that, we have used some reference standard for the calculation of their relative retention time as well. Because in case of mass spectrometry, if you're going to couple with the chromatographic techniques, so it may offer the identification of compound based on retention time. So we actually introduced this relative retention time factor as well for their absolute identification. So after development of the database, this is the strategy that we have used for the analysis of so many plant extract and the identification of particularly alkaloidal plant metabolome. So this is the first part where you can see that we have developed the strategy of the strategy based on tandem mass spectrometry. And the second part is nothing but the analysis of different medicinal plants. So once they analyzed and they all medicinal plant, the spectra were blast to the database that we have developed. And if you just look at these two spectra, this is the spectra of our standard, and this is the spectra of the same compound that we have identified in the sample. So this is the strategy through which we are actually generating a quick chemical fingerprinting of so many plant species, and particularly the analysis of medicinal plants as well. So again, you can see that, as I have mentioned, that this is the standard spectra of one of the alkaloid, and the same spectra that we can see in the plant sample. And you can see that this is the spectra which is actually being provided by the Bruker library. The currently available Bruker library is only giving one ion for their confirmation. However, the library that we have developed is actually providing the multiple ions for their accurate identification. So it's again a kind of database that we are developing is on is a priority in terms of like the features generation as well. As I have mentioned that after developing the database, we have analyzed a large number of plant sample, and this is one of the examples. You can see the Aconitum heterophyllum, and a large number of alkaloids were identified, not on the basis of their mass error, but you can see the retention times that we have observed and the drift. You can see that in the real sample, the retention time is quite negligible in comparison with the database that we have developed, which means that it can be further facilitated to the other laboratories for their identification, the accurate identifications of the compounds. Although we have not commercialized that database because it's still under construction, but I think soon once it's constructed, so we would come into the market and hopefully to provide the rest of the laboratories for the analysis. And I'm pleased to share that because the ACS has actually covered our findings on their cover page, just published last year again. As I have mentioned that we are actually using the similar strategies for the DEER applications and the chemical fingerprinting development of various species. So without going into detail, this is one of the example which you can see that we have analyzed 5 salvia species. We all know that salvia is one of the largest genus in the Laminaceae family and possesses a large number of medicinal plants. So we observed the distribution of very important metabolites in all this species and also developed the comprehensive profiling both on negative and positive ionization. This is one of the example related to not only the plant species, but the analysis of different agriculture fruits as well, which belongs to Cucurbitaceae family. We know that the cucurbits are known for their taste and medicinal importance. So this is one of the example where we have extensively studied the distribution of metabolites, secondary metabolites over 8 to 9 species of Cucurbitaceae. This is a very nice example I would like to present here because there was a case reported that there's a cross mixing of poisonous species with the herbal medicine. And, they proposed that probably because of the similar morphology of the dried material, because once the plant material dried, then it's quite difficult to identify that what is what. So you just rely on the third-party supplier that the raw material is actually the same which you are expecting. So there was a report that there is mixing of a poisonous species. So we just exactly work on that and try to differentiate that even in the dry form, if you have, then you, with the help of fingerprinting, you can differentiate the species from the medicinal species. So this is the just a model, after the metabolomics analysis, that you can see that all these 3 species is actually separated, although the one species, the cystrum diagram was the poisonous species, but it is well separated from the 2 other medicinal importance. The another high throughput methodology that we have developed few years before, back in 2014, is very much high throughput, I would say. It's just the use of laser desorption ionization. Because we are familiar with the GCMS analysis and LCMS analysis, and whenever there is a name comes from MALDI, the metric assisted laser desorption analysis, so there is always a restriction that only is useful for the large molecule samples. So we just modified some procedure without using the matrix. We used laser desorption. Excuse me. So we just use a simple laser desorption procedure for the quick profiling of various plant material. So what we have done, it's a very simple procedure and very high throughput. You just need to take a few milligrams of plant material, just need to powder it, because the dried material is quite easy to powder. So after just being sieved into a small particle size, which normally represent plant dust, because if you just the sieve like floor or whatever, you can see very fine particles. We just place this fine particle into the MALDI plate. Of these particles are actually sieved into different mesh sizes. So I'm not going to talk about in detail about the mesh sizes. In fact, you can see that the finest particle of the plant material is actually lying under the 50 micron size, and we just confirm through the AFM and other techniques as well. So once you apply this fine particle with the help of some organic solvent, a few microliter, 3 to 4 microliter drop of organic solvent, this picture is become like this. This is something before treatment. But once you apply a solvent of organic nature, particularly like dichloromethane or chloroform, so it just destroy the outer layer of the plant cell wall. And this layer is, once destroyed, and when you apply the laser as a source of energy, so then the metabolites come out and detected in the mass spectrometer. So this is the technology that we have developed, the protocol that we have developed. And you can see that with the help of the developed protocol, we have analyzed a large number of plant species. And you can see that the picture, the profiling, the chemical fingerprinting of each species is quite different from the other. And we were able to identify a large number of metabolites in that sample as well. How we can identify? Here is the example. You can see that this is the laser desorption profiling of the tobacco leaves. And we all know that the tobacco is known for the nicotine. And this is the lift experiment and the MSMS experiment of the nicotine standard. And if you just compare the same from the sample that we observed this exactly the same. So this is how that would actually identify a large number of metabolites by very high throughput screening of plant material. The method that we have developed is actually only applicable to the soft tissue like leaves. It's not, unfortunately, not applicable to the hard tissues of plant like fruits or roots, I would say. And that's why it's actually the people are using that. So this one of our collaborator, they exactly use the same protocol after the publication of our paper, and they use the laser desorption exactly by using the lichen species, because the lichen species are soft in the outer side, so they exactly identify. So a large number of citation was observed by using this methodology. So the second part, and probably, yes, is the last part as well. So is the use of, how we can use this sophisticated technique to address the issue of herbal plants or phytomedicines. So in this way, I would like to give an example of one of the product. This is the Lincus product, is produced by the Herbion, is one of the actually leading herbal company in Pakistan. The Lincus product is useful for the throat infection and the chest infection, and it consists of over 10 plant species. 10 plant species is quite complex syrup actually. So the company faced a problem of the raw material issue. There is one of the plant material that they are using in this formulation is Adota vesica. Adota vesica is a plant, again, is having an property to treat the throat infection. So they asked us that how they can resolve this issue, because they are relying the raw material on the third party, and sometimes they collect it by themselves as well, but they are not they're not getting the desired efficacy of the product. So they asked us that how we can solve this issue. So there is an example where you can see that the role of advanced mass spectrometry tools for the chemical fingerprinting and for this standardization. So as I've already explained you that the Adota vesica is one of the medicinal plant widely being used for the treatment of respiratory issues, and thoroughly available in India, Pakistan, and some other parts of Europe as well. So what we have done, our strategy was to collect. It was quite comprehensive study that we have conducted in collaboration with the company. We tried to collect the raw material from all possible sources. So these are the sources. You can see that from where sometimes they used to collect. Unfortunately, in Pakistan, we do not have a proper field where they can get the sustained raw material. They mainly rely on the third party to supply. So these are the places from where we have collected by ourselves and also collected the third party. So over 14 region across the country from where we have collected, identified them, that this is Adota vesica. So after necessary sample preparation, these are by the way, the exact locations of the. So after some necessary sample preparation, preparation, we analyze them using LCSI, MSMS, and you can see that the profiling of all these species apparently looks quite similar, and it should be similar, except the difference of some intensity of some secondary metabolites. So the first step was to develop an authentic chemical fingerprint of a plant species. So this is how that you can actually develop a database, and which can be used for the longer time period for checking the quality of the raw material. So right after that, we subjected these to the metabolomics analysis. The large number of metabolites, you can see that. I'm sorry that you can't see the name exactly here. There's so many compounds that we have identified, and we performed the different statistical analysis, and all species apparently looks quite close to each other. We perform a different experiment, including a heat map analysis, the hierarchical clustering, just to see the distribution of metabolites. And the metabolites distribution across the species were quite similar. So then we were confused that how we can declare that the which raw material. The first step is done, that the fingerprinting is done, but the another question, that which raw material is actually good for the efficacy or for the standardization of the. So then we analyzed the linker syrup extract. You can see that this is the linker syrup extract, and this is the standardized powder that they have actually for the preparation, and this is the one of the Adota vesica sample which we have actually collected. And we just compare the profile. You can see that these parts are actually, these are the metabolites who actually coming from the Adota vesica in linker syrup. As I have mentioned, that the linker syrup is a mixture of 10 species, so it's quite difficult to standardize these species, these 10 mixture species against each one. And again, to tell us that the which part is contributing from the plant side in terms of the bioactivity. So these are the metabolite that we have seen as coming from the Adota vesica. So we were able to identify these metabolites, and these were actually 4 important alkaloids. You can see that visicinol, visicinine, 7-hydroxyvisicinone, and 5-methyl. So these are the 4 alkaloids who are actually coming from the Adota vesica into the linkers, and they are actually responsible for the biological activity as well, because the activities of these compounds somehow is reported. So we just combined data with the reported data, and we were quite able to see. So that second task was to quantify those alkaloids in these species that we have collected and in the sample that we have. So we purified them with the help of recycling HPLC, and after extensive structural characterization using spectroscopic technique, including NMR, we were structurally characterized those compounds. And now those compounds were used for the quantification of these molecules into the species and into the sample as well, using the multiple reaction monitoring approach. MRM is a kind of approach through which you can quantify the compounds and metabolites in a complex mixture. A complex mixture could be plant extract, a complex mixture could be herbal product, a complex mixture could be biological fluid like serum, plasma, because in most of the bio study, the drugs are being quantified with the help of MRM, and these are highly sensitive approach for the quantification. So we use the MRM approach to develop the quantification profile, and all samples that we have collected were subjected to the quantitative profile. And when the quantitative profiles up, and we were able to identify that these species collected from these places, the upper hilly areas of Pakistan, were actually the right raw material to produce the herbal samples. Like you can see that the viscocin and visicinone were in the high range in those samples, so from 932 microgram per gram. So it's indeed the best raw material that they can. So after identifying all these things, the company was so happy, and they just started to collect the sample from their cell. Probably now they are making foam over there, because we don't know the exact reason that why these secondary metabolites are producing in high amount over there. It could be climatic effect, or could be the soil chemistry, whatever. But now the company is happy, and now they are probably getting the so this is the strategy that we have published, and now most of the people who are working on the herbal standardization, they are citing our strategy, because we identify the exact step-by-step strategy that if you want to identify, if you want to standardize the herbal extract, so what you need to do. The first step, you propose the collection of plant material. What's the objective? Obviously, to develop the authentic chemical fingerprint and to monitor the environmental effect. The second step here, to develop the untargeted metabolites analysis, and the objective, obviously, to identify the secondary metabolites. The comprehensive analysis, identify the marker component. Isolation, purification, spectroscopic characterization of marker metabolites. Obviously, it can be used as a reference standard for a longer run. So this is how that we actually propose the use of advanced tools for the herbal standardization, and the paper was published, I think, few years before in Metabolomics. These are the few examples of our research group contribution in the field of, again, natural product chemistry, although we are working very extensively in the biological system as well. But as this workshop belongs to natural product, so that's why I'm more focused on this. I think the facility has already been highlighted by Professor Ratia. So being a mass spectrometric in charge, so I now just added a one picture of few instruments that the mass instruments that we have. We have dozens of mass spec. But the very good thing that if you see that this instrument, this is MAT 312, is actually the oldest instrument that we have in our center, and it's a world record, because no one in the world that has such an old instrument and fully functional. So the old instrument, but functional as well. So this one is that credit goes to, obviously, to our beloved teacher, Professor Dr. Iqbal, because he's so kind in a way that to hire the skilled technicians and the engineers, and they are since long they are working to make all the instruments functional, and we feel really a pressure that all instruments should be functional, but at the same time we are happy, because this is the requirement and the legacy I think we need to learn, and the other institution also need to learn, that how to make a institution truly functional. So these are a few examples. These are the two mass pack that we have recently purchased, again from the Bruker. So I think with this, I would like to lastly acknowledge the students who are actually contributed in this work, funding agencies who are involved in supporting by one or other way. So with this, I'm very thankful to all of you. Thank you very much.
Clap now, clap now. You clap, it was muted, so no. So thank you very much, Professor Said, for giving us this nice presentation on metabolomics. And I would like to open the floor for questions. Any of you who have any question for Professor Said before I ask my question? So, Professor Said, thank you very much for your presentation. And metabolomics is an area of research in natural product chemistry, which is lucky in the African continent. There are some pockets of such, but we would like to strengthen this. And we would want to engage you and the people at your center in conducting a training workshop. First, at least to start with, online, and then we work out and how we can raise funds so that we can have a physical workshop, be it in Nairobi or at ICCBA Center in Karachi. Any question? Hello? Anyone online who has a question? You can type and accept the majority of them. They have just accolades, and they are thanking you very much for your nice presentation. Of course, in metabolomics, probably the most important part is on the selection of the plant. I mean, capturing the material you are going to study so that it does not change, so that you capture the metabolic profile of the species at that particular moment. So what is basically implying, depending on the season, depending on the time of collection, the metabolomic profile of plants of the same species varies significantly, may vary. So for purpose of helping, for example, a company like you were describing, worrying about the metabolic profile of the product they are supplied with. Since the plant synthesize these compounds for their purpose, for interaction with their environment, it might be variable. So how is it possible to use this metabolomic tool for purpose of validating herbal preparation, phytomedicine? Can you please just enlighten us on that?
Yes, actually, as I have mentioned that the herbal products are quite complex. The current situation, the current technologies that the companies are using, they're mainly relying on the cost-effective technology. Like they are still using the TLC densitometric analysis for validating the raw material or sometime products. They are using the IR, for fingerprinting of raw material. Sometimes they are using, if they are really have a good budget, so DNA as well. So the mass spectrometry is a kind of ultimate solution for a sample like herbal products. It requires a huge study first in order to declare a metabolomic fingerprint, I would say, using mass spec. As you have mentioned that the metabolites quite varying from season to season and from place to place. So this is very well true. And it's also depends that at what time you are going to plug off. So making all these parameters constant, once you develop a chemical fingerprint with the mass spec, believe me, it's quite authentic. What we are doing as well, because as you have seen that the compounds are being identified with the help of tandem mass spectra. So now we are developing LCSI MSMS library of plant extract as well, the herbal raw material as well. So once they developed, so in a longer run, so it would be quite easy and cost effective, I would say, to address so many issues related to herbal plant, or on their products as well. And I'm damnly sure that because I have not presented a work which is actually linked the chemical profiling with the biological activity, because the ultimate purpose of the product is nothing but the biological activity. The biological standardization is really important if you're going to talk about the herbal medicine. So the mass spectrometry, the tools are actually there where you can link the chemical marker with the biological activity of the product as well. So I'm very optimistic, and in my opinion that in upcoming years, because I'm also working with the I have an experience to work with the NAPRECA, which is a German herbal company. So I can see, I've just visited a few weeks before the company, so I can see by myself the company is totally shifted towards the high-tech mass instruments for their routine standardization of plant material and their products. So that is a need. Probably the other companies will come later on, but it would give you ultimate solution. So in my opinion, I think as it can address the aspects of issues related to the raw material quality, I would say, and the herbal product quality. So that's why the people are shifting there. Yeah.
So good, thank you very much. And there is another question from the chat by Madina Adia. And I don't know if you are seeing it in the chat, but she's basically one is metabolomics able to identify all the metabolite or compounds present in a particular plant species, or it only looks at the dominant metabolites?
Yes, actually, I have presented is all about the untargeted metabolomics. You know, that is the beauty of mass spec, that is unbiased. Because you can see all possible metabolites that is present in the plant extract because of its high sensitivity of the technique. So it's up to you that how you can identify that the spectra would be there, but sometimes the people don't identify. So the answer of the question there, yes, you can see all metabolites present in the sample, and it's again subjected to the expertise of a person who's going to analyze or prepare the sample or collect the sample, is very much subjected up to that expertise. But yes, you can analyze almost all types of secondary metabolites using mass spec, and we usually call it untargeted metabolomics. And you can see a major metabolites as well, or one or two metabolites as well. This is what we call targeted metabolomics. So both experiments can be done with the help of mass spectrometry.
So because I think you have this targeted and untargeted metabolomic profiling.
Yeah.
So how, of course, it is in principle, it should be possible, isn't it, to get to identify all the metabolites in principle, but it will be a very uphill task. But
yes,
targeted probably much better, isn't it true?
Yes, you are somehow absolutely right, because by using one technique is absolutely not possible to cover the metabolites, but you can cover the maximum metabolites present in the sample. And as I have mentioned earlier, that is very much subjected to the expertise of individual person, and which includes so many people, like including the collection of the plant material, the processing of the plant material, and the analysis of the sample, analytical mass spectrometry analytical technique that is being used, and the person who's going to interpret the spectra. So these all actually, if you are really lucky and you have all these a good combination, then you would cover a broader range of metabolites. So these, I mean, if I would say yes, which means that you need to have a good team working with you, and the full of expertise in their their respective area. So then you can identify the largest metabolomic coverage using mass spec.
Good. Thank you very much. Can you please once again join me in thanking Professor? Thank you very much.
Thank you.
We will continue to engage with you now that we have an agreement between our institution, between the University of Nairobi and ICCBS, between ICCBS and NAPRECA, etc. We'll work as a team in this Afro-Asiatic region of the world. We have very rich biodiversity, very rich folklore of traditional medicine. We need to work together and come up with solutions to help our society. We are very grateful. Thank you very much.
Thank you very much. Thank you.
So all the same, you can look at the chat from my brother Aman the cable is asking, are there Are there possibility you also collaborate with similar centers which have molecular bank and databases and get more number of database of compounds?
Yeah. Actually, there is a mass bank. There is a global bank, which actually where you can pool your spectral database. It's a free of cost. So the people are working in the development of a global database, and we are quite open for that as well. So if anyone is interested, so we can talk to them later on through exchange of email or through Professor Iqbal. So I think personally, don't have any issue for that.
Okay, good. Thank you very much. Thank you very much.
Most welcome.
Thank you very much. So now I now welcome Professor Justus Munyoki Mulwa, who is online. Can you please share with us your presentation, Prof? Professor Mulwa? For some reason, I know you are unmuted, but we are not hearing you. Professor Munyoki. Zoom sometimes has problems with. Hello, Prof. Professor Munyoki, are you? We are not hearing what you are saying. Prof, we are not hearing you. What could be? I don't know. Eh? Is there anything you can do to help him? No, is that?
Can you hear me? You can hear me?
Yes, we can, Prof.
Oh, okay. Can you see me?
No, we are not. Yes. Yes, we can see you now.
Should be able to see me. Okay, so I'm trying to project in a minute. Good. Can you see my presentation?
Yes.
Yes, we did.
All right.
Prof, we can see it. Could you put it in
Let me
presentation mode?
introduce it. It is in presentation mode already. Are you able to see it?
We see it, but
I've done so already. You can.
Yeah. It's not yet in presentation
It
mode.
is only in the presentation.
Oh,
Okay.
maybe you just
Okay.
go to
Now I'm out of the. I've
Just
now
go to
left
the-
the presentation mode. I got.
go to the very first.
Okay.
On that line where you're touching the beginning. Beginning at the top.
Oh, okay.
Up
Yeah. That,
again.
yeah.
Yes. Just click there.
Which I've done. Is it better now?
So,
Is it better?
Yeah.
it is not.
You can see it
projected.
at least, eh?
Yes, we can see it. So what you can do, Prof, this where you see click add nodes, that place collapse it to the bottom so that it can expand a bit. The click add
yes,
nodes
yes,
where you see
yes,
a click, that. Yeah, but we'll see when
Okay,
you do
let me
that.
ask you.
Yeah. The click add nodes collapse it.
Is it better now?
It is better. Let us just try. Though it's not showing.
We continue
us.
that way.
Just go ahead, Prof.
But you can see the slide there, maybe it's smaller, but you can see it.
Yes, yes.
So the participants who are present in this meeting recognize the professors present who just made their presentation this afternoon. I was following Professor Atia, Professor Syed, who have just done a very good presentation. It was really jargon to quite a number of us who are not in that area. So I'm going to make a few slides in a slightly different approach. The others are talking about all these kind of things, metaphorisms and all that. Me, I'm going to talk about a software area. I'm going to talk about the product business models, value chain. I know that the professor said you have talked about natural products. What are they? How do we extract them? What do we do with them? I'm going to just say a little bit as a professor of marketing that what you are doing is very important. You do extractions, you make all these products. The question is, when do they get to after that? My name is Professor Munyoki, Professor of Marketing, University of Nairobi. We deal mainly with the products. We help people in this country to find markets because we are able to produce. Production is not really a problem. Most of the time we are able to produce, but what many people tell us, including those in the field of medicine, is that they occasionally experience challenges getting markets for their products. And I think that's why I was asked to make just a few statements about that area. And I'll be focusing on the value of natural products. Why do we need them? I'll be looking at the value chain levels, a few slides about packaging, labeling, and branding, because that is a very important area. And then I'll say something, just a slide also about the business model for natural products. I will also mention something about intellectual property and maybe just give one or two tips about what we need to do in the area of marketing. First of all, natural products. We know these have been used traditionally and even commercially in the modern society. These are being used. We cannot deny that they have been used. They have been used to treat diseases. They have been used as food flavors and all that. Just how do we get them to that level? This is very important to appreciate the important role that natural products play in our society today. And as such, to be able to make these natural products available to the end users, we need to have effective, we need to have efficient systems, value chains. Value chains have to do with converting the products from the raw level at which they are to a level.
Sorry, Professor Munyoki, we were still seeing your first slide. So I think you click
so
on
it's
the
not
slide.
moving.
Yeah, it's not moving at all.
Okay, let me. It's not moving?
Yes, it's still on the title slide.
It's on the first slide, eh?
Yes, Prof.
Okay, let me just exit. I think I'll exit and then just come back. I will stop presenting.
Yeah.
Then I connect it again. Is it available now? Can you see it?
No. So, Prof, is it possible for you to send
Yeah,
it
let
to
me. Yes, I'm sending it to you immediately.
Just
Let me
right
send
now
it to you kindly.
So that
Just a moment.
No
I think
problem,
let me just send it to you. Yeah, we solve that problem. I'm sorry about that. Let me just send it to you right away, Prof. Just give me a minute.
No problem.
Okay, your email, kindly. Your email number.
Let me check.
Your email and Dr.
Okay.
Your email. Just tell me your email.
Okay. I'll put it in the chat. Prof.
Get over to just a minute. Just 1 minute. I really don't understand. Just 1 minute.
No problem.
Just 1 minute. Just 1 minute. Yes, I'm just getting there now. I'm checking. Give me just a minute. I'm almost there. Give me just a minute. Just a moment, I'm almost there. All right. Mhm. Much for gadgets.
Prof, maybe can we try it tomorrow or tomorrow first thing in the morning? Is it possible at 8:30? So, Prof, you are still muted because I know you have a class coming in 10 minutes. So you are still muted.
Okay, now that's okay. Can you hear me now?
We can hear you now.
Okay, let me just try. Let me
Yes,
just give it another try, a final try.
Because we can also do it tomorrow at 8:30 if you are free.
Unfortunately, I will not be free tomorrow. But let me just see whether it will come. If it comes, then we can decide. I know members are
Okay, good, Prof. Yes, yes.
Please let me be able to share. It is disabled. Okay.
Are you able to allow me to share?
I think you were
Says
out.
host disabled.
But now you are now you are
Now
a
I
co-host.
am. Okay. Can you now see it?
So it is the same. So what you do not put it on what is it, on presentation mode. Just leave it like that, and then every time, so click on the side, just like that. Click on that one.
Is it moving?
Yes. Yeah, it's better. So you put, when you are done with the slide, click on the side like that. It is better.
Okay.
Now, can you click now on the third one?
So are we there?
Yes. So
So
now
we
we
are
are
moving.
on. Yes, yes.
Okay. Okay. Thank you so much. Then I'm sorry about this confusion, but I didn't introduce this slide, the importance of natural products. That emphasized. That is where we are. And I think all of us know and appreciate the important role of this. So I was here. Are you able to see the slide? Slide number 4?
Yes, yes.
Natural product value chains. I'm saying value chains are important. Value chains will facilitate the availability of products, the link between those who produce them and those who need these products. And we need efficient systems. We need effective systems. And as we go about that, it is important to understand the players. Who are the participants in these value chains? How do they play their roles? And I think these are very important dimensions of this. And we need to ask ourselves certain questions. Where do the raw materials for the natural products come from? We have just listened to a presentation here by Professor Said about the extractions that take place in various parts of the world. So where do we get them? Who harvests them so that they are able to be used in those other processing as they do the value chain additions? Who manufactures them? And who buys the end products? Are they available locally? Where do we find them? If we wanted to get a product today, where would I get a natural product? Are they exported? And who exports these products? So as we look at these dimensions, we are really looking at the value chain, the power of the value chain in linking all these networks. A study done recently found that 65% of consumers of natural products are actually in the mean to level organic consumers. They are not really the big ones, not very small, but the mean to level. They buy a bulk of these materials. So how do we get the materials for them at their various levels? So there are various levels of the value chains, the extraction level, where we have to go to the forest, where we have to do the extractions. How is it being done? Harvesting is very important so that we make sure that we are harvesting the right ones, that we can verify, so that as they go through the processes, then we are sure that what we are carrying through the process are the right materials. Identification is very important so that we know that what we have harvested is the right material. It has been identified and verified. Women play a big role in these processes. Sometimes they have groups that are able to extract, but the other people won't just buy, won't just go to the forest and make small extractions. It's small bits, and they will sell them to individuals. Some will sell to manufacturers at various levels. So we are talking about all levels, including the individuals who take a lot of effort to do these extractions. That is very important. There is what goes on there. Then what do you do with those harvests that you make? They will go to various levels of manufacturing, both at primary level manufacturing, secondary level manufacturing, conversions and all that, so that they're able to make sense to the customers. And this is where marketing now begins to take place, that we are interested in how you do it. How do you package? What are the quality concerns? How do we check for quality so that we know that the cost, whoever buys them is buying the right thing? Whether you are doing it for the small manufacturers, whether you are doing it for the large manufacturers, the concerns are the same. We are concerned about the equipment that is being used to do these extractions and manufacturing. How do you store it? How do you ensure that the right temperatures, the right conditions are maintained so that quality is not interfered with? And then there will be the many distribution outlets, both locally, both externally. We find them in the supermarkets, retail shops, all of them being retailed. Concerns are coming here. How do we manage those concerns about the quality when they are there? Do we need to have supply agreements? Terms clearly spelled out about how they are being handled. How are the end users being used by the distributors? Whether they're individuals, whether they are corporates, it is important to have this in the value chain that is going to be effective and to serve the purpose. Of concern, members, is about the issue of packaging, the issue of labeling, and the issue of branding. This is very important because many of them may look like they're the same. Just how do we do it so that we make sure that there is that distinction that is created? What kind of material are we using? And we need to be concerned about the type of material, because that material can affect the quality of the product that is within that package. What is the cost of the package material that is being used? Might that interfere with what customers can afford, their affordability? There are environmental concerns. What kind of material are we telling people to use when they are packaging? Can they be recycled? Do we communicate? And you can see the image there full of writings. Do customers read what is written there? What exactly goes into that? Of course, that may be part of labeling, but we are just concerned about the writings on the package. How is it communicated? How do we maintain freshness in the package? That is done. How is it handled? The safety concerns and all that. So there are quite a number of things here that we are talking about, and we have listened to Professor here Said talking about the issues of standardization. Very difficult to have a standard approach to all these. There are a lot of variations, but all of it is important to understand. And that's where we come in as marketers. We are concerned about labeling. What information do you put on the package? We have seen packages that say very little. They don't even have a date. You don't know, this is based by what time. Some don't even describe the ingredients that have been used in that. They don't even say how it is used. There is no issue, something about, what about that package? Can it be recycled? What do I do it after use? When do I throw it? Those kind of instructions are very important in a good package. And I think these are important concerns for all of us. And thirdly, in this area, is important for us, both manufacturers and for us as marketers, to understand the issue of the brand. What is the name? We must move away from generic to brands. Strong brands will sell themselves, but do they stand for that which they say? You give your brand the name, you have a specific symbol there, even the colors there, and all these things come in here. To what extent is this be used in this phytomedicine area when you're talking about natural plants? How are the trend names given? Are they registered? What are the trend marks? So some of these are used as creators of distinction to bring about the uniqueness in what we are trying to sell, because we are dealing with many patients, people want to be healed and all that. health concerns, and people are concerned about that mark of quality. Does it stand? In Kenya, we talk about registration with various regulators like the KIPI and all that, but I think there is also the poisons, what we call the Pharmacy and Poisons Board, that requires certain standard materials, and they would like to see whether our products meet those criteria. So as a business model, let us go for one that involves all stakeholders, the users, the extractors, the manufacturers. They need to be there so that they are able to identify the value that they are able to add into that system. Every step in the whole process, right from the production in the bush to the time the product gets space in the shop, every step matters, especially to the customers, for those who use it. So it is good to have a model that is cost effective. It is good to have a model that can be sustained. I think this is the message we bring to all those who are in this area, those who are working on this. Sustainability becomes very important. There are many players in these value chains. There are researchers, there are scientists. What role do they play in this whole process? There are regulators, there are distributors, there are stockists, and finally there are the users. Each one of these and many other participants need to be clear about the role that they play, and they play that role in a way that is not only effective, but that is also sustainable. I'm sorry, I'm rushing people because we are also tired, and as I said, I was also going to join a class we just started, but I hope we are following. Are we together, or might I be talking to myself? I've only about 3 more slides, just highlights.
We are together, Prof.
We are together.
Oh, I'm
okay.
going to talk about.
Okay, lastly, let me just mention about the IP. What you have gone through since Monday have a lot to do with IPs. Presenters here are talking about what they are doing, coming up with very good innovations, groundbreaking. Just how do they protect those from others who may be there? So we want to encourage that all the scientists, all the researchers who are working in this area of natural products, please try to find a way of protecting your innovations. Get intellectual property rights so that other people using that will not be able to do it without getting permission. There are a lot of good things about IPs. I will not be able to go to that, but whoever gets an IP stands to benefit a lot from others who may want to use that. First of all, it protects your idea so that no others are going to go that direction without your permission and all that. Licensing, there are many ways of doing this, including licensing others who can use your idea, but they use it with your own license. So it will be given to the creators of these persons who have come up with the ideas so that they are protected. And related to that is the issue of patents. Patents will give the owners, the scientists, this legal right so that they are able to exclude others from using the same ideas or even selling them. So I think this is an important area that we all need to be familiar with, so that as we go about the various things that we are doing, we also need to go the extra mile to make sure that we protect what we do. And these are important concerns, and I've been listening, following since mid-morning, the various presentations, and I saw a lot of issues that have to do with IPs. So a patent is a legal document that is given to an inventor, the scientist, whoever, so that you protect that subject matter that seeks protection. So in our own country here, Kenya, in other countries may have a difference. For those who are working in Kenya, please look at our constitution. What does it say about property rights? What does it say about those inventions that when you're extracting them and you're saying, this is my idea, what does the constitution say about it? What does the Pharmacy and Poisons Board require? What must you do so that you are safe as far as that, as we place our products on the shelves, in the pharmacies, in chemists and all that? Have we complied with the drug regulatory authority requirements? Let us look at Consumer Protection Act, because we are telling them to buy our products, they are safe for use. Have we complied? Let us look at like the Kenya Protection Act. Of course, there are several others. I'm just saying that let us look at these various frameworks so that we are not caught on the wrong side as we do these things. So finally, because I'm a marketer, I want to raise some concerns here. Why are we doing all this? Why are all these people doing the extractions? Are they driven by profit motive, or do they have other motives? Why the effort? Is that a major concern for people in this area? Do they expect to make money from the products that they make? What care do they have as far as their customers are concerned? I want to say that in marketing, we believe that our main driver is to satisfy customers. We must make those products that meet customer expectations. That is when we get satisfaction, that when we use your product as prescribed, we are able to get the benefit, to get what we wanted to do. That is what we can call the utility level there. And you, the producer, need to be able to get a return in terms of a profit or whatever it is, so that you are not just there, there must be the motive. That is very important. And therefore, we need to understand our customers. We need to understand our markets. Markets are different. The market in Kenya is totally different from the market we find in other places like Pakistan and all that. There may be a few things that we may need to change. Markets in Europe are different from the markets we find in Africa. We need to understand our customers. What are their characteristics? What are their income levels like? What are their gender characteristics, the age, demographics, and all that? It's important to understand those. This, gentlemen and ladies, if we are able to understand this, it will help us so that we are able to come up with proper market mix considerations. It will be able to touch on the product itself, the package sizes, the package color, and all those. It will be able to focus on the pricing structure, the pricing that we do for our products. That's why we may have segments, market segments. A segment in Africa will be different from the segment in other parts of the world and all that. And even within Africa, the Kenya segment, if you look at Kenya as a market segment for the various products, it may be a little different from the market in some other place like in Ghana. A few differences here and there, and we need to be familiar with this. We need to understand how we communicate that which we have so that we are able to get the customers using the language that they understand. And then we talk about how we distribute effectiveness, the right product, the right place, and all that. These are important considerations. And lastly, it's good to understand their customers, your capacity to develop them, especially the large customers. If you are supplying a product in Kenya, do you have the capacity to serve the market in Kenya, or do you want to serve a small segment within Kenya so that you are able to reach your capacity and be able to guarantee the supply so that you don't supply next time you are not able to. It's important to attend trade fairs so that you get more information, trade fairs, whether locally or abroad, wherever it is, so that you are up to date with what customers need and want. Good to connect. There are many associations that are dealing with these kind of things. What do they say? How do they help their members to market their products? And these markets have different requirements. let us be familiar. Your products are meant for the European market. What are the requirements? You are dealing with a product for the commercial market here in Kenya and other parts of this session. What are the requirements for the product that is being marketed within the commercial industry? And there are many other markets that we can talk about. Ladies and gentlemen, thank you very much. I think those were really rushed, and but I think I've done it. Thank you so much for listening. Maybe we back to you.
Thank you very much, Professor Munyoki. So thank you very much, Prof. Anyone with a question for Professor Munyoki? So, Prof, thank you very much for giving your presentation. This is one of the area many of us in the biological and chemical sciences are not good at. And we are not good at selling things. We are not good at promoting things, etc. And we will continue to engage yours and Professor Mary Kinotti's office so that as we develop our phytochemicals products, will be properly guided. Thank you so much, Prof.
Okay.
Thank you so much.
Okay, thank you so much.
Yes. So, Prof, I know you are going for your class. Thank you so much, Prof.
So thank you very much, members. Thank you, everybody. I think I want to wish you all the best. I'm sorry I came at the end when everybody is tired. But nevertheless.
Understand these things.
Okay, so.
Yes, thank you, Prof.
Bye, be blessed.
Okay, good, thank you. So thank you all very much. So this marks the end of our session today. Tomorrow is the last session of our summer school. And we will have, today we had a lot of NMR. Tomorrow we'll have something on GCMS and LCMS by Professor Anthony Gachanja. And as you will see later, Professor Anthony Gachanja is a trainer for the Royal Society of Chemistry on GCMS and LCMS. Tomorrow, please come early than today so that we expect to finish. So the people in the bus, if you are not here by 9 o'clock, we will finish with the online people and we'll be going home. 8:30. We start 8:30. So tomorrow 8:30. Tomorrow 8:30. Okay? What's it? 8:30. Okay. Thank you so much. This, we are not going to be confused, okay? We will be here.